shell, then fibrous coat, then you get the nut, which is very soft. You can cut with the knife. Normally nut, you know that you need a special device to cut it, but this is just with a knife you can cut it. Okay, and they are taking half of the piece with a little bit of leaf and lime. It's very important, putting it here, chewing it, and you can think that people can chew for 10-15 minutes and whole thing swallow, nothing spitting out. And people are taking initially 2-3 per day, but then its addiction is coming and increasing. Gradually it will increase. Like see, you can cut easily with the knife. So this gradually increasing, and you can think about 10-15 minutes per chewing things, you can chew it. People are taking 30-40 per day. So you can think how long they are chewing it and swallow whole thing. When I met this individual, so when I met this individual, he's a farmer, you see that blood is coming. Okay? So what is happening, I came to know that he can't stop it to chewing it because addiction is very much there. Even blood is coming out. And I have written that for the first time when, I don't know. Okay. So first time user can really feel the effects that can cause eating koi, which is locally known as koi. Chewing of koi causes many serious complications from changes in heartbeat, blood pressure, bringing sensation in the mouth, mouth areas, leading to many serious complications including cancer. Okay? And but I am telling that 100 patients from Meghalaya are going to CMC Vellore, and some doctors came and they, this is just quote from the newspaper, taking a lot of unprocessed, beef and pork and other things. So there are so many factors involved in it for the cancer. But my focus is just not can you use cancer or not. Okay? And you know that in the nut, what happened, particularly in the raw areca, alkaloids are very common. These alkaloids, alkaline, alkylene, guanacine and guanacine, so these are the alkaloids which are there, and you see that you are taking little lime, so the alkaline pH, and in the saliva, it converts to nitrosamines, and most of the nitrosamines are carcinogen, okay, and which you are chewing it, and then gradually swallow these things. So these nitrosamines which I have given, any of it, more detail I am not going to teach you because of time, but if you want to know something, we can discuss afterwards. So now this is the areca nut induced cancers pathway. See, ROS, reactive oxygen species, hypoxia, it can create, it can also it can affect. All these pathways there, my mainly involvement in the securing and GST, who plays role in these areas. We started and gradually, so I'm not showing each one, just showing the result. That glutathione is one of the small tripeptide compound we have, having areculin-induced DNA damage in mouse and bone marrow cells. So influence of glutathione is there. We will not discuss detail about these things. Each one I am showing and published one after one in some papers. Areculin oral administration is very common. Peritoneal, if you give, it will not induce in cancer. That also we have seen in mouse model. And N-acetylcysteine, in a short form of MAC, which is a precursor of glutathione, if you give it, it can protect areculin or say not induce damage, which is coming in cancer later. Then ROS, we have shown that yes, partial effect of this nut definitely mediated through ROS, reactive oxygen species. With two different protocol we adopted those days. One is with superoxide nitrate, another is creating anoxia and treat the cells with the nut extract. Then we started moving outside the lab and collecting the samples of blood from the market, from Barabaja. And then what we have seen, heavy RAM chewers showed significantly higher DNA damage, delay in cell kinetics, P53 expression, and lower endogenous growth hormone level. So that means if these are the parameters, you can screen people and can tell who are in danger for these kind of habit, that chewing of nut. And this is the molecular level. We have seen that 9p21, chromosome 9 short term, second band, first subband, where p16 gene is there, whose loss is 70%, 40% loss and 30% methylation. Okay, so that data we have shown it, and then, but now I will show you actual story here. While I was doing these things from till 95, 96, 97, that time, a lot of my colleagues are telling, Dr. Sir, the nut cannot induce cancer because people are here chewing it for so long, and I don't think nut is inducing something else. I don't know, I think, because already WHO is telling that nut is carcinogen and its component. Definitely processed nut, the effect is much lesser because your alkaloids are volatile, so it will be lesser effect will be there. But then I told that, okay, I will prove it that nut alone can induce cancer. How early we can detect? So these are the two main questions which I followed afterwards. And then by the time I already established my contact with the hospitals, particularly in GRIMS and Nazaret. Nazaret I prefer because they have a very good track record. Even 50 years before, whosoever come and given it, that record also you can get, even the tissue. So they have very fantastic system. Okay, you can collect a lot of information. So I started collecting samples, human samples. Initially I thought that I will make primary culture, which is very tough to make a human tissue and setting culture. It's not so easy. So in a good lab, 50% the success rate. When I started, it is 15 to 20% success rate, but I started. And I made chromosome of human that cancer biopsy samples. From that is a G-bending, thinking I will see any fixed translocation between two chromosomes, which is very common because if you are students you are teaching, studying cancer, you see that 922 for the CML, 814 for Burkitt's. So lots of cancer having specific chromosomal translocation. So I was thinking of that line, but then what I had seen, this effect. You see this effect, every chromosome were sister chromatids separated. Okay? Initially I ignored, but then I saw that in some slide it is very high. And then I started counting it. I have found that 30-35% cases, you can see prematurely separated sister chromatid. Although you are preparing this kind of plate in presence of colchicine, which is a metaphase arresting, even then they are separated. So this one actually attracted my attention. Afterwards, in mouse, those who are familiar with the mouse chromosome, they are all 40 chromosome acrocentric, but see, each sister chromatid separated. Okay? When you treat mice with this kind of round, we have not extract. So that we have seen it, and then I started gathering information. Now what I have seen that this is called genetic instability or chromosomal instability. And you know, genome instability is a hallmark of cancer. So this you can find it, and I found some paper in Nature in 1998. This is I found in 1997, 98, around that time. Then I started reading it, and I find that people are suggesting that yes, genome instability or chromosomal instability is a hallmark of cancer, but nobody knows the cause. And that is the time when people started working on it, that what pushes them, the instability. People are telling that probably checkpoint genes problem creating this kind of things. So in that line, I started contacting people, mostly in US, and I got offer in Sloan Kettering and Columbia. Then I moved there for at least a couple of years. There I worked. And what they have done, HCT116 colon cancer cell line, which is a stable cell line, all checkpoints intact, they are actually knocking out MAD2. MAD2 is metaphase arrest defect, which is a checkpoint genes, 5 exons. So exon 2 that those days siRNA technology not introduced, okay, early year 1999, 2000. So they knock out exon 2 by the neomycin cassette, and after knocking out, then confirm it by southern blot that yes, you have a knocked out cell. Only thing is that MAD2, every gene we have a double dose, but one single knockout is survival, double knockout is lethal. So single knockout, they confirmed with the southern blot, with the single knockout cells. And after that, what happened? That in the single knockout cells, you can see that here, you see that this is knockout, this is the wild type, this is knockout. In the knockout cell, you can see within 36 hours, what happened that so many plates having 8N, 4N, because you have a 2N, divide 4N, divide 8N. So many cells are in 8N, whereas in wild type, they are still 4N. 8N is just negligible, but here significant amount level is in 8N. So that shows that cells were being passing very fast, and which you have seen that sister chromatic separated means they are not, stay long in metaphase. They are passing metaphase going to anaphase haphazardly and distributing this sister chromatid not uniformly in the two pole haphazardly, and that's why number of chromosomes also will be deviated from the actual 2N number. So then what will happen? When you will see that cyclin B associated with CTC2 kinase activity, the students may know that when cells are in metaphase, you have cyclin B and CDK2 top level, okay? So what is happening that in the wild type, that is called MPF, metaphase promoting factor. So this is from 12 to 24 hours they maintained, okay? But in the knockout cells, at 12 hours peak, but then gradually it is diluted, okay? Because the cells is moving from one stage to another, okay? So that's it shows like that, and here you can see that since the cells are not for long period staying in metaphase, what happens when you are cell in metaphase? If you do culture in a flask, when cells arrest in metaphase, you will see cell become round, and if you tap it, they will float. Okay. So in this case, what happened in the wild type, all are round, okay, rounded and detached from the flask. Don't disturb it. Again, they will settle down. But in these knockout cells, all are round, okay, and they will not detach. I mean, so they will detach, but they will not sit down. So you can collect those things, and if you expose for longer periods to nocotazole or colchicine, both are same, then you will get polyploidy. Okay, multiple nucleus in the one cell. After this, what happens that if you see the normal thing, this is metaphase, sister chromatic. In metaphase to anaphase, sister chromatic will be separated. Now this separation, you need a protein called separase who can separate it. Cut the protein cementing substance. But separase cannot act because securin bind on it. So the point is that this is called APC, anaphase promoting complex, who will dissolve the securin, separase released and cut. Now the point here, the securin level we are measuring, and what is happening because in knockout cells you are looking sister chromatic separated very early, that means securin must be lost. So what we have seen here, this is normal and your knockout. Normal knockout 6, 12, 14, 16, 18, 20 hours. You see in the normal cells, the securin level intact till 24 hours. But in the knockout cells, securin level goes down. Okay? So securin is the one who releases separase and it cuts the sister chromatid and they are separated early so that you are getting this premature anaphase separation, which is called precocious anaphase. Then what's happening now? That kind of effect leads to aneuploidy, means chromosomal number imbalance. That we want to see. But here you can see by using fish with chromosomal centromeric colored probe, you can see every chromosome, you can see 2 dots. Okay, that means sister chromatic separated. Normally you should get 1 dot, but there are 2 dots separated. And here you see that white one is the wild type, okay, and this one is knockout. So two different knockout cell line. You can see that number of chromosomes in human it is 46, but they are deviating 42, 43, 44, 47, 48. So deviation is much higher in knockout cells than the wild type cells. Then see here very clear, chromosome 1, 7, 8, 9, 11 and 12. Randomly you picked up. One is red, one is green. You see that these three cells were 2 red, 2 green. Chromosome 7 green, 8 red. So 2, 7, 2, 8. Perfect. But see there are some cells where number of green and red very high. Okay, so that means aneuploid is very much there. And then what happened that we again select 4 different cell line of knockout, and this means 4 different cell line means in a mouse, but 4 different mice. Okay, or say sorry, this is cell line, still cell line. Next I will go to mouse. So these are the 4 different cell line, and what you have seen in HCT116. 45 is the model number. Chromosome number is 45. I told very stable cell line. So in the wild type there are chromosome number 45. How many cells deviate? How many cells deviate from the 45 number? It is 11 and 15. But in the knockout, you see that number is still 45, but plenty of cells deviate from the 45. 32, 29, 30 and 50%. 50% deviation. Model number change from 45 to 47. So it's very clearly showing that yes, knockout cells are really unstable. Then people ask that yes, you have shown cell line, but you have to show in an animal system. Otherwise they will not believe. So in the mouse knockout, so in small category they have a knockout, MAD2 knockout mice. So from the knockout, the embryonic embryonic fibroblast. You can do the chop the embryo and set the culture. On the embryonic fibroblast, see this is the normal mouse chromosome, and in this MAD2 knockout, you see every sister chromatid is separated. Okay, so that it shows that see in the knockout cells, you have 30, 36, 57% cells showing such kind of effect, and percentage of aneuploidy is same thing like 30, 36, 57, 57% showing 75% cells aneuploidy. So the percentage of anaphase, percentage of premature anaphase separation and percentage of aneuploidy cells, they are linked. Okay, so it shows that yes, it is the effect of that thing which we published that time in Nature, and then they showed the MAD2 knockout developed lung cancer within 2 months. Coming back to that, now I want to show you that NAT alone can induce cancer, which I was telling that. Now, these are my students who did their PhD from this department. What we have shown that this NAT plus line, whenever it is there, you see that almost 35% cells showing that premature separation. That premature separation is this, premature separation. And this is only NAT. So my point is that there is one more question is coming. That will leave it. Oh, I think it will stop. So one more question is coming that lime, what is lime is doing. So if you don't add lime, the effect is slowed down. That means alkaline pH is very important. So what happened that in case of lime, the number of premature anaphase will be reduced say around 18%. But with lime, the effect is much higher. Now, this is the effect, and ultimately you will get aneuploidy. 37, 38, number is 40. So you are getting, and see these things, you are getting polyploid. This is in the mouse. And what we are doing, we are feeding with the water. Okay? Mind it, this is, we have seen in the bone marrow, not the direct tissue. You are feeding mice with the water. A mice normally takes 6 to 7 ml water per day. So you mix the RAM extract as such that every mice is taking 1 mg per day. Okay, for 2 months. Then next 2 months make it 2 mg. Next 2 months 3 mg. Ultimately 4 mg, and we are continuing till 360 days, almost a year. And what we are looking at here till 180 days. this much. But we have gone beyond that, 240, 280, 300. I'm not showing all the details, but he is showing it very significantly increased. Ultimately, what is happening, along with that, all these checkpoint genes, Aurora A, B, MAD2, BAB1, everything we have seen, the effect is very much there in the your northern line. And if you see the P53, everywhere northern line P53 increases significantly. Then if you see sequeling, level also increased. Then if you see parks, also increased, but I don't want to mix up right now. Let's concentrate on P53 and sequeling particularly. All the effect we have seen, Western as well as those days RT-PCR. Then you see that mouse which are given 240 days onward, then you see the bulk stomach, huge stomach bulk, cancer induced. Now the point is that in mouse, you cannot do oral because you are giving through water which they are sipping and quickly pass through mouth and esophagus, rest in the stomach. So stomach affected highest degree, okay? And therefore you get the stomach cancer. This is normal, this is cancerous, and we have done histopathology showing the cancer kind of things. And then randomly you picked up normal, only not on line, okay? Only not 300 days. It is still in the early carcinogenesis, dysphagia, but not on line, full-blown cancer, carcinoma. So when line is not there, cancer induction process is slowed down. Okay? So that's the thing which I can tell. Now, when you have seen in the mouse, this is which we published in BHC Cancer, then we are thinking that let's see, same thing happen in the human or not. So again, my students got to VH, they requested people to give their blood. They brought by sumo, we have given, their breakfast. Otherwise, they are not ready to come in the early morning. So 8:30, they are coming here. And those days in my lab, people are not coming because they told that they don't choose that. So somehow we manage. And then let's see. The beautiful thing is that people are not suffering from cancer. They are taking not 30, 40 per day, going to the village and farming and all those kind of work. You are collecting blood. This is normal human metaphase plate, okay, which we are preparing in our thing. But see these people, they have this effect. All the chromosome. It is there in the blood. Mind it is not direct tissue. You are chewing on your mouth, going through the alimentary tract, coming in the blood. So from the blood you can see the effect. And these were, and see the number of chromosomes. Normally 46. These people are in polyploid. And they are moving. And I am telling you that one of the students who is with Partha Madhukar in Kalyani, she collected samples from his around village, and she told me after 5 years, 3 died for different cancers. One is breast, one is something else. Means the effect is very much there in the system. So that's why I told that this can be a good screening marker and biomarker you can add. Just take the blood, within 2 days you can give the report and you can tell, instead of nut, please take carrot. Antioxidant, otherwise problem. So this is the thing, and see that non-chewers, 8 I collected, and almost 18 samples, heavy chewers, randomly, and see everywhere you can get sequeling level up. In non-chewers, sequeling level very low, which is usual. Very low sequeling level. P53 also goes very up. So these two definitely a very good protein marker to look into it. But now the question is that it's the same thing. Sequeling level, we have done immunohistochemistry from the biopsy sample. These are oral cancer, these are esophageal cancer, and this is the mice we fed 240, 300 days, and tumor. Everywhere you can see sequeling level is very heavily expressed with the immunohistochemistry, with the antibody binding on it. On the slide, you can see on the slide from the patient sample as well as the mouse. This we publish in Oxford Journal, that in human, this kind of things we can see. Now the question is that, which is the last part of my talk, is that how and why sequelin is increased. If you take NAT, sequelin level goes up. If you go in literature, you will find that people attempted to find out its point but failed it. Okay? No methylation, no mutation, nothing is there in sequelin, but why its level goes up. So that's the question, and I am showing you it's in mouse. 0 days, 60 days, 100 days, 180 days, 240 days, and 300 days. See how the sequelin level goes up. Okay? And also with the Western, you can show 300 days sequelin level significantly more. So this we have shown it. This is all immunohistochemistry data. The sequelin level goes here and 300 days. Now, when I found the literature, then I found that one thing is very common, that whenever gene expressed high, they have some epigenetic modification at the promoter level. Okay? And you know in eukaryotic chromatin, there is a wrapped chromatin with the histone protein. And in histone, you have H1, H2, A2B, 3 and 4 in your nucleosomes. So what happened that the 10th part of H3, histone 3 I studied, not no other histones, and H3, there are positions where some kind of epigenetic modification taking place, post-translation. So what happened that, like say lysine 4, K4, trimethylation, K9 acetylation, K18 acetylation, all in lysine. Arginine also there, but I have not taken arginine, all lysine I have taken. So there, either methylation or acetylation is very important. Some are favoring transcription, some are suppressing transcription. Like K4 trimethylation, favoring transcription. So if it does, then K4 trimethylation should increase in sequelin promoter. Okay? K9 acetylation and K18 acetylation should increase if it favors your transcription. So that, from that level, we started looking at it. And what happened, I have done H3K4, H3K9 acetylation, 18 acetylation, and K9 methylation. K9 methylation suppressing transcription. So if sequelin level goes up, means more transcription, then trimethylation should increase in K4 and methylation K9 should decrease. Whereas K9 acetylation should increase. That's the thing. And if you see this immunohistochemistry, each one, it shows exactly the assignment. That K4 trimethylation increased, 300 days, see, from the picture is very clear. Acetylation increased, K9. K9 methylation decreased from here to here. And K18 acetylation also increased. This is on the tissue, on the treated in the stomach tissue of the mice. Okay. Then there is another important loop is there. What is happening that I think I should go to the next slide. Then you will understand. Normally, what happens? We have a cyclin D and CTK4. When cell cycling started G1, this P16 will off, and then cyclin and CTK4 join. Then they have a kinase activity. With which molecule they will do the phosphorylation? RB, retinoblastoma, which is a very important protein, which is binding with E2F, is a transcription factor. Normally, so what is happening? They will phosphorylate RB. Once you phosphorylate RB, E2F released, enter into the nucleus, bind on the DNA, and so many target genes will be expressed. And if you see the target genes, MAG2 is one, then sequelin is another one. Okay, so one thing is that sequelin expression goes up, means RB must phosphorylate, E2F should be released, then sequelin goes up. So this is the point, and that's why what I am showing this slide, that everywhere in 300 days, RB phosphorylation goes up. So RB up means E2F released, and E2F going up. With these antibody in the tissue, we have seen it, and that means that yes, RB phosphorylation goes up. So that means if you take NAT, your RB phosphorylation going up, E2F released, and some of the target genes, only one target I focus right now, sequelin, but now subsequently we'll do more, then this is definitely going up. This is, I'm showing you the sequelin genes structure, okay? Because we are designing primer. Now we will do the chip, showing this histone modification. And for the chip, I have selected 4 primers. This part is promoter. In the core promoter, and further to upstream, one is downstream. So from class 6 to -874, in that region, I selected 4 primers. I created 4 primers, okay, and then we have done the chip assay. This is the one you have seen it. So this chip assay we have done to see the chromatin. Now what happened? This whole line, you can see 4 primer, 1, 2, 3, 4, and this is H3K4, trimethylation, acetylation, 18 acetylation, 9 methylation. Now once you have seen it, now see what is happening here. This is, the black one is no antibody, and this is the antibody. Now what you have seen it, this is control, 100 days, 300 days of NAP extract feeding. In 300 days, if you see all these 4, primer 2 showing maximum retrieval. And if you see the scale, it is 1.4, otherwise all in 0.8 scale. Okay? So maximum effect retrieval of chromatin you can see in the core promoter region. Okay? And in the core promoter, this is what's the region? -83 to -192. So that region we found maximum retrieval. That means the close to the gene, the changes is most top level. Okay, that change. Naturally, question comes: fine, you are showing your gene, but you please show me some in heterochromatin region. So we selected chromosome 11, where is no gene is there, called gene desert region. So we created primer from that region, and we have done the same study in the gene desert region, where you cannot see that kind of epigenetic modification taking place where there is no gene. So this is a kind of negative control. And also we have done normal histone. Okay, all these effects, normal histones are same in control 100 days, 300 days. So histone protein are same, but its modification varies. Okay, and so this is positive control. So all those things we have shown it, and then each primer, after that when DNA retrieves, we purify and then go for real-time PCR. Then we have whatever the things we have defined, submitted for sequencing, just to see this 4 primer region, the sequence match that thing. Okay, so whole thing sequence done, and already I told primer 2 is the most in that area where the highest level of epigenetic modification taking place. And all the genes or proteins which is favoring for methylation or acetylation or suppressing methylation or inducing acetylation, all these enzymes, N-methyl transferase, acetyl transferase, demethylase, all these things we have seen, and they are matching with the effect. Okay, wherever methylation more, methyl transferase is more. Wherever methylation down, demethylase is more. So that's why it's down. It's showing real time as well as it's showing. It shows with the immunohistochemistry. Okay. So now one point is there. We are taking NAT for our enjoyment, no problem, you can take. But the point what ultimately leading it, if you take NAT, and if it NAT increase global level of H3K4 trimethylation increase, which gives an indication that NAT relax your chromatin. Okay? So more NAT if you take, more relaxed chromatin. Now it is indicating, now in the next I showed it, that yes it does it, because what happened that here, untreated and ran line, stomach, bone marrow, esophagus. Okay, you are doing it, and then you extract that nuclei, and then take the DNA. It's a little different way of DNA extraction, not the similar way. Then after that, if you do the gel, now see that, after that you have to treat with DNAs, because if the chromatin relax, they will more cut by DNAs. Okay, so if you do the DNA treatment with 2 different concentrations, then you see that this is normal DNA without any treatment. DNA treatment, this level goes up. And if you have a marker, you have seen 2 kb down that level goes up. That means 2 kb, and that means your DNA's fragmented DNA of 2 kb size is increasing after RAN and line treated one. Okay? Same thing, this is stomach and bone marrow show the same thing. Esophagus, it didn't show that distinct. Point is that in esophagus the exposure is very brief because mice is taking water, it's just going very quickly passing. So esophageal tissue are not exposed to that longer period. But stomach definitely showing it. Then what we have done, we pack the gel and elute the DNA and purify it and do the PCR of securing it. And then what we have seen, everywhere in the DNA treated, RAN treated, and DNA treated, whatever the fragment you are getting, see the securing level goes significantly high. We have also selected one housekeeping gene, GAPDH. GAPDH level also goes up. So these things are telling that yes, indeed chromatin relaxed and it increased the amount of DNA of the active area, active site of the chromatin, and many of the genes goes up. Okay, and that's how this particular model that you have not in line. Okay, and in mouse we are giving extract mixed with water and pH 9.8 by adding slight lime, and then when you are treating, this is the compact chromatin. Now chromatin become relaxed, and all these modification, time iteration of K-lysine and K9 acetylation all increased. Okay, and that's how we see the effect. So which we published recently, Cancer Cell International, and then this is also published in Mutagenesis in Oxford Journal, that DNA hypersensitivity. And these are my students who did all this work. Some are from geology, but from rest of these things is Dr. Rangat from Gujarat, and from nicknames, there are a couple of doctors are there, funded by DST, DBT uses SAP program, DST FIST program, so lots of expenditures involved in it. Some of the students are right now postdoc, either in University of Pennsylvania, some in Columbia, and many are well placed even in this country. So thank you very much.
Blood screen people are not bad. They can be scientists too, and here we see the example like how good is doing in, and it's very good for our future. So the session is open for questions. If any questions.
Okay. Question T10.
Okay, T10 you can ask a question. So we can move to the next. CSR ITIT New Delhi. After PhD, he moved to healthcare department on ETBS Institute of Information Technology Delhi, and currently he leads the computational health science research group at Ashoka University. So I welcome Dr. Rintu Kudam for his lecture. Please.
I just would like to thank the organizers for inviting me in advance. Okay, just to start with my talk of the title is open source scientific computing and also open source tools which we can actually use for discovering biomedical research. So what I have for today is to just briefly introduce my lab, which is currently at Ashoka. So we typically span from our research from cellular ecosystem to public health, where typically we use the computational aspects of the research. Major theme that actually encompasses in our group is to actually help research scholars, and then also use the same learning to advance the computational thinking in the public health domain also. And we primarily focus as a long-term goal to contribute into the field of computational spatial biology. And also, apart from all the helping and others and doing own research, primary emphasis is to actually facilitate the what we call as patient-centric ecosystem in across India. So the whole idea that my lab actually works on is from one of the very nicely illustrated by Charles Offray, which actually talks about how we can actually leverage big data in health settings. So in healthcare, typically we come across from, like, if you actually ask from cellular ecosystem to the public health, or in fact the other aspects of the ecosystem, we have high volume, high density of biological and also clinical and environmental and also life science, lifestyle informations, and which are collected from not only from the single individuals but also across many individuals. And to do this whole activity, you typically require a lot of participation of different stakeholders. It starts from if you look at the public health researchers, so you also require public health policy people. You require representative from health science services, drug developer, pharmaceutical industry, and ICT, which is information communication technology companies. And also you require clinicians who really actually see the patients, and biostatisticians who really make sense of the starting from the small data to large scale data set. Then you also require biobank to store those bio specimens, and also you require patient advocacy group, which is typically look after the how the patients are going and how to make their life more easier when they are in the disease condition. And also you require experimental biologists who actually do go beyond the what do you call it, just a public health perspective, but also you go into the molecular mechanisms to find interventions. Then you have like us, which are typically bioinformaticians who actually help biologists, and also computational biologists who are typically go beyond the typical bioinformatics analysis. They go to machine learning and AI. And there are special class of people out there who are like genome scientists who typically look more deeper into the genomics of individuals. So if you actually look at the whole the training cycle that actually goes into training that kind of interdisciplinary people, you will have to have some frameworks. So typical frameworks that we also follow is of having a system medicine perspective or system thinking perspective when we are actually training or in fact doing the whole work. So you can just imagine a laboratory which is doing a lot of molecular studies. If they are coupled with the what we call as ICT platform effectively, you can actually connect effectively the genetic and the environmental or in fact biological clocks, which is actually governing the health and the disease condition of individuals. And this has been very nicely put in this particular review where how we should train people and of different backgrounds who might be interested. For example, a clinician might be interested in doing more computational work, or computational biologists who would like to go into the clinical settings. So when we actually see the the structure in at least in EU, how it works is typically you have a research interest which comes from it's a researcher, but there are something to do with the clinical utilities, which is at the end of the other spectrum. If we don't actually connect effectively this bridge, we will not able to provide patient care very effectively. So the main goal is that each of the component really is driven by training. So if we don't train well, so we will not able to achieve the goals that what we call something called as a precision medicine. So what typically the environment that we push forward is to do collaborative research, which is more to do with systems thinking perspective, where you actually always care for the patient-centric solution. Then after that you come to the population-centric one. Then we require clinicians who understand the informatics also, because there should be some people who become a bridge between the clinicians and also the scientists. And you require community effort. Community effort typically to do with how do you sustain that kind of training ecosystems for longer run. So because if you have ever seen a research career, it is typically spent from 20 years to 40 years. After that, you have to train also skillful people who are not in the core domain, but in the intersections. And education and training becomes a very important component of the collaborative research. And there are many stakeholders there which is vital when you are doing research. So primarily my group actually focuses on different verticals. So as we see in the how the collaboration is very important with right partners and like-minded people. So right now we actually have four different teams. First is on the computational cancer pathology perspective, where we are actually using deep learning algorithms or advanced AI algorithms on digital pathology, which comes from H&E or in fact your what you call as multi protein capture at tissue level. And you can see that we start not don't do it starting with our own cohort, but we help others. The team right now is from Jamia Mia Islamia, where he is leading the group. And we have other partners from Maulana Azad, AIMS Delhi, and we have from RPI, MS Ranchi, and BHU Banaras. And on the main perspective on the computational genomics, we actually help a lot of people across the NCR region. It's spanning from single cell biology to the more of an evolutionary biology perspective. Then we also cover on the computational public health, which is the primary focus of our the center that I belong to, and also the Ashoka University. Its primary goal in the biomedical domain is to actually facilitate public health people more researchers, because we will find computational genomics people or bioinformaticians with a good number in India, but we don't have people who are very much highly trained on machine learning and other public health. We also closely work with private entities like hospitals, apart from the public hospitals like Max Healthcare hospitals, and also we started working with the startups also, which are like they actually cover mostly the the troubles that we get faced when we have to partner with a lot of multiple hospitals. So my healthcare typically now actually encompasses around 75 private hospitals across NCR. Then we also have something to do with futuristic little bit, which is on digital health, where we are actually trying to develop technologies or existing technology, use those, how we can actually make accessible the research or the knowledge content to the very remote part of our in India. This typically we call as a folk computing, which is like the compute is for the folks which are very remotely located. So just a snapshot, what we actually do is also AI-assisted cancer pathology, which is already shown. This is an ongoing project we are under the Indian Cancer Genome Atlas project, which is led by Professor Sashidhara. Then we also do a little bit on the molecular side, cell biologist part, where we actually try to actually predict and find ablations. So if you see in the first what you call as the figure, there's no ablations. But if you see in the second one, there is a ablation that was induced, and it was able to actually quantify with the machine learning and the deep learning algorithm that how much was the displacement after the ablation. Then also we work in the area of antimicrobial resistance, where we do at the both sides. One is on the clinical isolate perspective, and taking the clinical aspect, we also wanted to understand what is the genetic basis of their resistance in those clinicals, so that we can provide surveillance systems across India. So the main framework that which is I have been discussing, and in fact I have been trained on, is on the philosophy of open scholarship. So basically what we believe in is that there should be open data, there should be open source softwares which we should use, there should be open source hardware which can be used for our research purposes, and open access. On those, the most important part that we have is something to do called as open notebooks. Open notebooks are something to where we can share across researchers, and we can actually get feedbacks from other researchers who are experts most probably. When you are a beginner, then you would always love to have some support, not from your within lab, but also across the internet. And one second is the citizenship science, which is to do with how you involve common people for doing whatever you are doing in the lab, or in fact in the clinical settings. How you make sure that you also advocate for the public. And most important, which I feel like, is the equity and diversity and inclusion. So I think when you come across communities like open science community culture, the primary thing that we learn as a part of the community is equity and also diversity, and respect each different backgrounds where they come from, or the level they actually have to start with. And mostly to actually again facilitate the open educational resources which can be useful for some of the scholars who are very new to a particular area. So when you actually look at the ecosystem, that primarily the research that is driven is, at the end we actually see the publication. Okay, the publication is the end product that we envision. But there is a lot of hurdles that actually comes when you are starting a scientific journey. You will typically don't have any idea with the, you will have idea about the data, but you will not have an idea about what are the things that are available to accelerate your research. For example, if you don't have a coding like the programming skill set and the tools that is already there, which can make your life easier and in fact do more faster, that path from the beginning to the end, which is the publication journey, it becomes very what we call as expensive in the sense of your time. So, for example, in India, we spend almost like 5 to 6 years, or in fact 10 years also in research time. It's all because of we don't train researchers or in fact scholars in the domain of computation, because that time can be reduced drastically. Because if you have to do a Western plot and if you have to quantify, if there is a automated script that is written in Python, which is the backbone of image, which is you typically use click, that can actually take almost 20 minutes when you start in the beginning. But if you write a small code, that can actually quantify your data like in seconds. So that's the advantages that you get, and also you get more time for your reading and doing experiments. And if you actually start the whole journey, it's just, as I mentioned, that you will start with a research idea at the first place, then you data planning and designing that is undergoes with the research topic you've chosen. Then you go and if you are doing a public research, then you go to the public, then you collect data sets. If you are doing experiment in labs, then you actually go perform molecular biology what you call as experiments. Then after you collect the data, then it comes to the data processing, which is effectively what we train people on the computational perspective of it. Then the second thing comes the data study and also the analysis. So if you're actually doing anything on the research, everything is quantitative. So if you don't understand basic statistics to a level where what we call as you can advance machine learning algorithms, today's day it is very difficult to publish also in good venues. Then once you've done the whole study, then you can actually go for the data publication and access, and how you actually preserve those data, that you can make the data available for others so that they can accelerate their research from what you have actually ended. Then that is what we call as a data and also the reuse principle. So let's start with something why we should actually learn a programming language itself. So most of the people actually think the life of a informatics person which is doing biological research, it's very easy to do, but it is not so. It's actually there are a lot of things that actually also comes into the bioinformatics or information, because they need to also advance themselves with the technologies that are moving fast. Like you in an experimental setting, you will have new devices that will come, or new instruments will come, which you have to also acquire and learn for accelerating your research. Likewise, the same thing goes with the informatics person. So what actually makes us when you have learned a simple programming language? It is to do with you actually become a better biologist, or in fact you become a better researcher. And most importantly, what I feel is we have developed with our training something to do with call as biological thinking that we have, but we lack something to do with computational thinking, and which is a little bit different from each other. But once you have computational thinking along with your biological thinking, it really makes your life easier in designing, or in fact seeing your own data with a perspective which is not being seen at the biological thinking level. So this is not a new thing that is recently came out, like machine learning and AI. This has been there from ages actually. So that's what I will try to tell you guys, that how the informatics has been very much, or in fact the computer has been very much effectively used by the developed countries, and we also now in a position that we can actually get computing more rapidly in our society. So this is a very nice article as well, which was written by Werner Gilbert in 1991, where he actually mentions the knowledge that we have at that point of time. It was so much that if you don't have a computer literacy, if you don't understand computer, then you will be lag behind. So that was a time like in 1991 that they were actually talking about. And also, the Sidney Bryder also mentioned in 1995 is that computing was not only going to be essential tool for biological research, but would also provide models for analyzing complexity. So it's like the amount of data that time used to be generated in NIH was humongous. So that's where he also felt the emphasis of how computing can be really useful in that point of time. So I'll just give a very brief, journey history about how computing has been an essential part in biomedical science. So typically it started with cytogenetics, as we have a lot of professor in this domain. You can see that it started with the name very early in 1970s, where you started using computer to do your analysis very rapidly, and also the algorithmic thought process that has been envisaged in 1993 of the modern bioinformatics perspective. Then you have in 1998, which was talking about how statistical analysis and along with the computing can really help cytogenetics. And in around 2021, there was a nice article by Bayat, which actually talks about how informatics is very core to not only understand biology but also in medicine and also maths and also computer science. So this is one of the fields that actually bridges all the different domains. And likewise, as the biomedical research came out and more advancements were done, so people started realizing that bioinformatics can become a very important what is a component in the clinical settings also. And that in 2014 there was the advancements in the computer also make a lot of the things easier for the computational biologists, is that you can do more work done because of the advancements in the hardware. So what I will catch up on right now is just for the students, which is to what are the things that you should be aware of, at least at the computing level. So I never started with my Python programming, but I started with R, because there was a you have advantages. Like when you are a biologist and if you are wanted to enter to the computing world, it's very important that you understand intuitively. Intuitively in the sense it's more of like a R is more of like a calculator. Like if you type in the interface, you will find that if you type 1 plus 1, you will get 2. But it's a little bit difficult to write in Python, that whole perspective. But nowadays we have advanced interactive development environments where you can do with Python also. But I will always recommend any new beginners to start with everything with R. Then we also do something like we don't give them data which is where they don't have any understanding, because for a biologist, you should have a biological data set to start with, where you understand the problem on that data itself. Now, the only task is the computer is that making that thing faster, which was not previously. Then there is a very nice software developed in MIT in Julia. So this is very fast, like faster than your Python and also R. And I think students should actually really start using Python, because this is the future of computing that will be the scientific computing. And this is also very intuitive like Python, but powerful like beyond Python and also the C++ itself. And there is one more is there, which is like for people who want to actually go more deeper. This is called Rust. So Rust has been very much used in bioinformatics settings, because especially genomics, because we have a lot of data set which is coming at the whole genome sequencing level, and the data set is very big. So you require very effective programming languages also to do the process very fast, at the same time at the cost of the time and also the compute infrastructure. So just to give a more flavor to it, that I will just talk about myself when I started my whole journey, so that you guys get motivated. So right now till 2012 to 2021, so I've been actually when I started my scientific journey, I started with this open culture, and that was 2012, where internet was very accessible for research scholars. We could do Google and everything and learn and get resources for doing a lot of the learning part. So my journey started first with one of the community activity that I did with the International Dream Challenges in 2013. Then I started involved with the Dream community since then. Still I have a community engagement with the Dream. Then apart from that, I moved across to other biomedical domains also, which is from the FDA, which is we call as precision FDA challenges, where you get biomedical challenging data set where you have to apply computational methodologies to solve the problem. So what you get out of from those challenges is to do with you actually learn from the experts. So typically in the community such as that if you ask like something, I don't know this, how should I go, they really help out with, what are the things you should do before it. For example, the same learning from there, what I do is, when anybody comes, like in our group, or, if they want to actually learn something, so we start with something very basic prerequisites. So prerequisites, for example, you should have a, little bit of maths, which is like linear algebra you should know, and you should know a little bit of, principal component analysis, then you move to the machine learning rank. So we don't teach someone directly the machine learning in the first place, but some little bit prerequisites that is required. Or for the computing part, we just train them with the R perspective, but finally they migrate to the advanced computing languages. Then also in the whole that tenure, what I actually got the exposure is since I did my PhD from IGA, Institute of Genomics and Integrative Biology. So there was a lot of demands that comes every year. Like there are dissertation people comes. So they come with a very different background, and that really gave me an opportunity to actually know how to train kids who are very new to the field, or some are computer science kids who actually really want to do biological data analysis. So I had the luxury of getting students from IIT Madras and also KIT University and VIT, BEATS, Finale, and they have a very different background. They have always come from the technical domain. You have on the other side, you have Naipar Korbada, who is like very pure, pharmacy background, but they don't have an understanding of computing. But also they would like to go for the computing. Then you have a Gautam Buddha University, JP, then Solan, then also MIT University, and there are many were there. So around like 22 people I could train during my PhD tenure. Then apart from that, also I contributed into a lot of the workshops that actually really helped me the differences between when you train someone in the very close environment, like in the institute, and training people which are very new, like in a workshop setting. So this actually resulted into one of the nice association with one of the member which I am. It's a reproducibility for everyone, where actually I was a part of the eLife as an ambassador. So this website contains a lot of the resources which I will be actually showing in the last. That you can start your journey from by just getting to know about what is reproducible research, what are the things they are for experimental biologists, what are the things they are for bioinformatics. Then there are other community activities also we have done, which actually highlighted in very reputed journals, where we started with everything to help the community without expecting the rewards. So one of the things that you learn also in the when you actually associate yourself with the diverse background and diverse people, you also get rewards somewhere down the line, but not necessarily immediately. And that is very fruitful when you are doing community services. And those are also led to of my own ideations, where the things were very much unorganized. So this was we developed in during my PhD tenure, which is called as ProMetaDB, which is the proteome level metadata. So if you look at the proteomics databases, they are not very structured like how you see in gene expression in transcriptomics dataset, or in fact genomics database which is there. So we developed something to call as you have a very scattered different data sets. How you actually bind them, so that the researcher can easily access the proteomics datasets like how we do for transcriptomics. So this was the technology stack that we developed, which is like you use R at the backend, and also something called Selenium, which is Python based. And those learnings typically actually resulted into something during my postdoc. So this was a project which was from the Principal Scientific Advisory from the Government of India, where they had a plan for like different cluster nodes across India, and I was the in charge for the Delhi cluster for the project coordinator, where I used my learning during my PhD on the open source scientific computing languages and also the open source tools. What we actually came up with is something we call as genomic surveillance of COVID-19 variant with language model, which is typically on the NLP side of the computer science, and also use the machine learning as on top of that. So what are the things that we developed? What we actually call is like you train a lot of what you call as right now you know about ChatGPT and all those. They actually comes from something you learn the pattern in the text. Likewise, we use the same methodology to learn the pattern in the DNA. Okay, and you use the machine learning on that, where we can actually say that or before 2 months ahead, we will actually tell that there might be number of cases might be increasing. And this was published in Frontiers. So the typical framework actually we work with. So you use the same open source scientific tools to actually get your automation of all the data from the GSET. Then after we do the GSET one, then we do have a lot of Python script and R script at the back end, which actually do automated QC and also annotation of the data. Then you started with only 2.8 million sequences to start with the spike genes. Then you use machine learning to actually predict that how you can actually get the predictions for the case loads. So one of the most challenging stuff that if you actually ask how the ChatGPT works, ChatGPT and the other language models have a very basic principle is that you have a text, then that needs to be converted into numerical space. That numerical space actually goes to the database and it finds a similar nearby answers. For example, if you ask the ChatGPT something, it will actually give you very relevant information. How it does? It's use your question to get a numerical representation, and that numerical representation goes to the most likely answer that is available in the dataset. Likewise, the same thing that we apply is that for the spike gene, we actually learn the structure for across 2.8 million sequences. Then we got the numerical representation. And the numerical representation is very important because if you are doing machine learning, you cannot do machine learning on top of text or the DNA sequences. So this was the framework that we do, and that was the predictions we could actually predict ahead of 2 months prior that what will be the case story in a particular country. Okay, so this was post my postdoctoral training where I contributed with a lot of the public health people started with, which recently published in PLOS with tropical diseases, where I did a little bit on the how R can be useful for doing non-linear modeling of the what we call as remote sensing data and environmental variables with the daily case studies. And this is also one of ongoing manuscript which is in communications, where we actually see that how pre-PCOS and polymethylene are the very intermediate what we call as phenotypes for polycystic ovarian syndrome, which is primarily from the what we call as cohort from the single cohort from India. So here also what we have done is very quickly. So basically the scientist and the endocrinologist was a friend of one of the AIMS collaborator that I had during my PhD. So she actually told about me that he can actually help you with the machine learning and all those stuff. So he comes with the data in, from Kashmir to Delhi, and I go from Ashoka University to AIMS, and we do the analysis in half an hour. So he was so happy that he could actually from the data and everything that you actually do, it can be very quick when you have a right partners. And the same thing actually we went rigorously for multiple rounds, and we actually after almost it was 4 months to actually compare publication quality figures. So this was the one which I was telling you about the oral cancer that typically we are working on, which is to develop the AI based methodology to detect oral squamous cell carcinoma subtypes. And this is for the students who are actually interested in this particular learn by themselves. So this is the open resources that is freely available, which is called as reproforeveryone.org/resources. So everything is in the what you call as site. You can use freely. You can use them to train other people once you understand the content. And it comprises of many stuff. It comprises of introduction to the reproducibility. Why reproducibility is very important. How you have to manage your data set. Data can be of your experimental data, or it can be of a computer or large scale data set. And there's something to do with electronic led notebooks, which is typically when we write notebooks in the labs, that might sometimes accidentally might get misplaced or it might be lost. So the electronic notebooks are very useful for experimentals. And there is okay, I have just almost done. So then you have something to call as protocol sharing for experimental persons. If you are doing some experiments which someone already have done, you can just get the right protocols from this side. Then reagent sharing similarly, then also you have some bioinformatics related contents which also give you a very starting point for your research. Code for data sharing contents, then you have a data visualization which is primarily for what kind of visualization you should use for your manuscript publications. Then how you actually do a publishing, the whole ecosystem itself. So the most important part which I feel like I was not a computational biologist or I now am a faculty in the Department of Computer Science, but I don't have a degree in computer science. So that was one of the very what do you call it? To motivate you guys that it is nothing to do with where you start, but what you want to become in the future, that is more important than anything else. So I am from Majuli, which is like a place district in Assam. Then I was born there. Then I came to Guwahati. I did my class 10, 12 till Guwahati. Then I moved to Bangalore for my BSc UG studies in biotechnology. Then in Odisha I spent my 2 years in MSc biotechnology. Then I moved to IGB for my dissertation. Then there I actually got very good mentors who were like Dr. Tapadesh Shetty and one is Rishida Swar. So these comes from a very diverse background. One was from an MBBS background who was actually doing computational medicine, and the second was was a computer scientist, science person who was willing to actually interested in doing biology. So because of them, I think I feel that it was a very easy journey for me to start with. Then also apart from that, I got a lot of mentors who are really helped me in at least enriching my whole what you call scientific journey in the process. Right now, I have a couple of 5 mentors here at Ashoka who actually constantly help us in growing because I am an early career researcher. So it's the support for an early career is very important in a system because you can't write big grants. So the big grants actually comes with them, and where they make us co-investigators, in which we also have a lot of energy, but they don't have that much of time, where we have more time to actually execute the projects. And within 1 year, so I have able to have a group of people around like 50 of odds. But these are the current people who are around like 32 people who actually working with me in the different projects that is there. And because of both of them, actually, I could able to accelerate the one of the projects that is there. Thank you.
Thank you, Dr. Putam, for telling us about all the tools we can use for our research. Now we are running late for the session, so we can go for next lecture, and question can be asked in the tea time.
Yep.
And the next lecture is from Dr. Preeti Singh. She is working in life science analysis department, and she is engaged in making the life-saving drug more effective by by her approach. So we can go directly for a lecture because we are running late. So
talk about immuno-oncology, which is my one of my passion area. And so this is what they say about the cancer, that if you use Windows, get virus, and use OS X and get cancer. And because we all are well-educated people and we know what cancer is all about, so we can say that people who joke about the cancer have no sense of humor, because the hardest tea to swallow is reality. So let me share some real figures with you guys. As we know, cancer is the leading cause of cell death worldwide. Every one death among 6 is happening all over the world. It's because of the cancer. And this figure is going to be tremendously high by 2040. This is what WHO says. And one of the major reasons behind this scenario is simply because we have aging population all over the world. In India, we don't understand this because we are the youngest nation. But think about it, after 20 to 30 years down the line, we are going to have an aging population, and age is a major factor to get any disease, and if it's cancer, it's even worse because we have limited number of therapeutic modalities available. So finally, we hear a good news recently that 14 out of 14 patients who were suffering from rectal cancer could be cured. And it was because of immunotherapy. So we need to know what immunotherapy is, and we need to get enlightened what immune oncology has to offer. So this study was recently published in a very renowned journal, ETBS. So let's understand what it is all about. So as we know, the major remedy for cancer from the ancient times has always been surgery. You just take out the tissue which is cancerous. Then later, as a mankind, we learned about radiation therapy, that you give localized radiation, can kill tumor cells. Then late 90s, we learned about chemotherapy. These are cytotoxic agents which selectively kills tumor cells, but of course they have side effects, so we have to use in an optimum concentration. So we have plenty of chemotherapies agents, like in hundreds or something like that. And then later, people were investigating, we developed molecular tools, cell biology tools, we learned about the mechanism, how cancer happens. So we learned about the targets, different targets like different proteins, inhibitors. So we developed those kind of agents which can selectively target those proteins. So they are called targeted therapies. I'll come to it later. Recently, from last, it's not been more than a decade since we learned about immunotherapy, and we all know the inventor of this stream got the Nobel Prize. But it's so, although it's not been a decade, but more than 3,000 agents are under clinical trial who are focused on immuno-oncology and immunotherapy. So this itself tells about the volume of significance of this kind of a stream. So altogether, whether targeting immune components or cancer cells targets, we have plethora of targeted therapies. But in order to have these targets, we need to know which cancer is expressing what kind of tumor, a specific antigen or tumor-associated antigens. We call it TSA or TEA. So if tumor is expressing, suppose HER2, as we know, triple negative breast cancer, we can target it using the monoclonal antibody against it. If it is estrogen receptor positive, androgen receptor, BRAF, then again targets can come from immune components like CTLA, PD1, and so on. So, but the real challenge is we do not know which agent will work for which cancer and for how long, because therapy resistance is a big challenge for cancer therapy. So people are trying to combine different approaches, like combine chemotherapy with targeted therapies, or do surgery first, or do the neoadjuvant first to give some chemotherapy and then give the radiation and so on. Immuno-oncology is nothing but you boost the immune system, because as we know, our immune system has the inherent capacity to deal with unwanted cancerous cells. So looks very interesting. Let's share. So in order to There are 2 FDA approved, although several are in pipeline. But when talking about T cells, we need to be very careful because we should know what we are talking about. Because it's a very thin line. So if there is any overexpression, overproliferation of T cells, it will lead to autoimmune disease. So this is the major challenge, that how to create this balance. Another fact is antigen to T cell interaction is a very complex process. So as we know, there is signal 1, direct interaction of T cells through MHC to the antigen present T cells. Then there is signal 2, CD28. And if you look at the overview picture of these different co-stimulatory signals and co-inhibitory signals, you can get an idea how complex this whole scenario is. So what are the major reasons for exploration of immunotherapy? First and foremost, it harnesses the power of immune systems, which can potentially prevent mechanism of escape resistance. Combination therapy are more likely to elicit the response and increase the response duration. And immune checkpoint inhibitors are addressing only one, though it's very powerful, component of the immune system. And promising data with combination of immunotherapy and another chemotherapy or targeted therapy can enhance more immunogenic cell death. So let's understand how these cancer cells escape the immune system. So this gives a brief idea. What happens is, in tumor microenvironment, there are various immune suppressive factors. They could be soluble factors which don't let those immune cells to function, such as T cells, B cells, and they recruit other immune suppressive cells like T regulatory cells, myeloid-derived suppressor cells, and apart from this, they also have ineffective presentation of tumor antigens to immune cells. And of course, T cell checkpoint dysregulation, and we have gone through previously. So again, just to give an overview, what we are talking about, this is the complexity of the whole immune suppressive environment, where cancer cells downregulate MHC expression, then fast ligand is overexpressed to promote apoptosis in immune cells, then T regulatory cells are present, and 2 type TAM. They are major culprit of immune suppressive function. They secrete various kinds of factors like TGF, platelet-derived growth factor, TGF-beta, IL-6, and so on. So this is what we are talking about when we talk about to get rid of immune suppressive effect of tumor microenvironment. Their major focus area while dealing with the immune suppressive nature of TME is MDSC, as we know, but it's very complex. There is a subpopulation of MDSC and we do not know whom to target and how. So this cancer immunity cycle itself gives us several areas where we can target, and we have been targeting it, like chemotherapy, radiation targeted therapies. Then at the second step we have vaccines. Alpha and gamma looks very promising. CSF colony stimulating factor, which recruits macrophages, anti-CD40, TLR agonist. Then of course immune checkpoint inhibitors, CTLA4, CD27, interleukins like IL-2, IL-12, anti-VEGF, CARs, and so on. So recently, researchers who are working in this area of immuno-oncology, their major focus is to block the other co-inhibitory molecules apart from CTLA4 and PD-L1 like LAG3, TIM3, KIR, TIGIT, VISTA, or activate the co-stimulatory molecules like OX40, looks very promising, 4-1BB, several clinical trials are already going on in this, CD27, IGOS. Another way is to block the inhibitory molecules like IDO. We will elaborate on IDO. It's very interesting. like which are neoantigen based. Then we also know about adaptive cell therapy like CAR T cells, TILs or T cells, signal inhibition like MET inhibitors, PI3 kinase, cytokines, IL-2, IF and gamma, or epigenetic modulators and microbiome modification. So these are different areas which is area of active research. So I am sharing this for sake of young investigators. If they are looking to build their career, these kind of areas can be very promising. So let's talk about IDO1. What it does, how it works. As we know, IDO inhibits the T cell activation by tryptophan catabolism. So if there is more accumulation of kinorrhea, T cells and Tregs cannot function. So this IDO blocks this IDO enzyme, so it gets rid of immune suppressive effect of kinorrhea and T cells can function. So one of the most talked about IDO inhibitor is ipecadorstat. It is a reversible competitive inhibitor of IDO1 and it is in the clinical trials. Other IDO1 inhibitors like BMS 986205, PF 06840003 and BGS 5777 are very good because they can even cross the blood-brain barrier. Then idoximod. It is already celebrated molecule because it has already got the FDA approval for melanoma. Other promising targets are to get rid of escape mechanisms targeting JAK-STAT pathway or loss of existing tumor immunity. Then we can utilize innate immunity space like toll-like receptors, RIG1 receptors, intracellular DNA sensors, C-gas and SPINT, or other T-cell engagers, T-cell bispecific, like which can block two sites using the one antibody. So this is also very much appreciated by the researchers and several molecules are already in the trial. Then pegylated IL-2 with anti-PD-1 is showing good response in the clinic. CD38. This molecule has drawn my attention and I am also utilizing this molecule in my model system. So this is the promising for mechanism of resistance to anti-PD-1 therapy. So it can be used in combination treatment. Then of course angiogenesis inhibitors, PARP inhibitors, IL-10, IL-2 pathway utilization and several personalized cancer vaccines are also showing promising. Just to have a quick look on adoptive T-cell therapies, as we all hear about adoptive T-cell therapy here and there. So three kinds of T-cell therapy, either TILs, like you take it out from the blood, manipulate it genetically and give back to the patients, or T-cells or CAR T-cells. Both of them are showing the great promise. Then innate immune cells like NK cells or macrophages are also being researched a lot to be used as adoptive T-cell therapy. Looks very lucrative, but most of the combination regimes use tumor microenvironmental approach and they increase the tumor shrinkage in the selected group of population. So they don't cover, they don't show good response to broad population. So we need to stratify the patients which can fall for this kind of treatment. So this is the main challenge with immunotherapy, that 50% patients which show response is amazing, as we saw in rectal cancer tumors. And where 50% of the patients, they are showing even adverse immune reaction. So this is the major challenge area, that you can't drop it, that it's not working. You have to counter the challenges, how to counter all these immune suppressive microfactors and make it work. So there need to be biomarkers for proper patient stratification. We need to have more effective and less toxic treatment modalities. We need to understand the response and resistance mechanism in each tumor type, what is happening inside, prioritize the clinical trial and tissue acquisition for new adjuvant settings, find alternative sources of biomarker analysis that's already going on, like cell-free DNA, emphasis on umbrella trials. Umbrella trials, I think we all know, this is where two patients which have different genetic mutations, therapy will be targeted to the genetic mutations. And we need to move beyond the single biomarker rather than IFN gamma and MSI. We need to have more kinds of biomarkers and we need to combine them in a certain way. So, if I put all these things in the monetary terms, then just one PD-1 inhibitor is projected to go up to $90 billion by 2040. It is already sitting around $30 billion. Just one. And if I talk about total immune therapy challenges, then in monetary terms, it is projected to cross more than $130 billion. So why am I sharing all this with young investigators? Because these are different pharma giants which are dealing with this area and trying to deal with these kind of challenges, how to make immunotherapy work which suits to every patient. So, like you name it, Bayer, Bristol Myers, Eli Lilly, Merck, Novartis, they are all the major players in it. What does that mean? It means it will create that amount of career opportunities, job opportunities, or even startup opportunities, like you can start your own lab and start building a career, trying to deal with one challenge. But the major thing is, these kind of jobs and career opportunities will go to only those hands which are highly skilled, which have good analytical ability, and it's not one-day thing. It needs a lot of patience and a lot of research. So, in this line, at LifeSense, on one hand, we are providing the precision guidance to the cancer patients. We are have collaboration with several local hospitals, and we are trying to create spheroids. So that takes an account of not only tumor microenvironment, but tumor immune microenvironment. And then we treat them with different therapy and a combination available. So beforehand, a clinician knows which therapy is working to what patients. So that's the personalized front. On another front, we are upskilling different biomedical scientists so that they are fit for the industry. Because as we know today, most of the academic researchers, they are not fit for the industry. Because industry demands different things. Although they are very talented, they have the right things, but it's not fit. So what we thought, we will bridge this gap of academia and industry. And in this line, we have MOUs with several universities down south, Meenakshi, then D.Y. Patil, and at least 7-8 universities we have a close collaboration with, and we look forward for more people to have a collaboration to enhance to make it work. So, thank you so much. Thanks for listening.
Dr. Singhal and Dr. Chaurasia trying to bearing with us because there are little subtle adjustment that we have to do. Anyway, that's part of our experiment. We all do science, right? So when the result does not come straight away, you got to bend your finger. So this is how we got, this was a negative control that we tried, it worked beautifully, right? So thank you, Dr. Singh, for a wonderful presentation. We thank our former previous speakers, Dr. is here, and Professor Chatterjee, who made a wonderful presentation. I also thank the audience for bearing with us. Now what we will do is, we'll continue as the schedule is. There is a parallel oral presentation that's going to be starting in the community hall, and we'll continue with the parallel session of oral presentation that will have a break, intermediate break. That is a tea break. Okay, so we take that as a tea break. So I request Dr. Singhal and Dr. Neha to continue. Dr. Hiranjit also is here. So you may have very extended session, so you'll have to somehow adjust. ठीक है, आप coordinate कर लीजिए, no problem.
Already finished with the Maya and Gupta. Now second one is the Gagi Sen. So I request Gagi Sen to please present our work. In 10-minute time, like you finish within 8 minutes.
Chair, please bear with my voice. My voice is sitting.
Then that's why we're giving 8 minutes.
I'll switch to my time. Respected Chair and other participants and other delegates who are present here, a very good morning to one and all present. Today's topic, that is phytoprofiling and dogging studies of ficus rootfield to elucidate its anti-cancer potential. This work has been carried out in the Molecular Genetics Laboratory and the Bioinformatics Facility Center of University of North Bengal. It's an entire teamwork. I am representing my team. So as we all know, the phytochemicals are gaining importance because of its less side effect compared to the modern drugs. So in this scenario, the ficus, which is associated with our religion, culture, as our national tree, this particular genus is a very ancient genus of around 60 to 80 million years old with 850 species. Some of the representative species have been depicted here. Ficus pumila, which is a climber, Ficus bengalensis, Ficus religiosa, and Ficus elastica. This particular genus has got a elaborate text in the ancient text regarding the medicinal importance. The multidimensional effect of the ficus, which can be seen as an antioxidative agent, anti-inflammatory agents in the treatment of scabies, ulcer, migraine, cough, diarrhea, anti-diabetic properties, etc. Now, why ficus rootfield? This particular species, Ficus rootfield, it is known as monk people, which is very underrepresented in the ficus literature. When we search the literature, this particular species is having a morphological similarity with our religiosa. If you can see the leaf, you can see the Ficus religiosa leaf or the people tree. You have seen it has got the leaf tip is pointed and elongated. In the case of Ficus rootfield, it is more or less cordate and wavy. So people mistook this Ficus rootfield always as religiosa, and you can say hardly any literature is available on this particular species, that is the Ficus rootfield. The taxonomical position is given. So the outline of my study is given here. So what we have done here, that is we have gone for both in vitro and the in silico validation of the anti-cancer properties as well as the antioxidant and antimicrobial properties of this particular plant. Previous slide, this one, right? Just an interesting fact about this Ficus rootfield. The Ficus rootfield starts its growth as an epiphyte, and here the first diagram you can see, it is growing in our campus on the eucalyptus tree, and later on in the older age, it grows at the root into the ground and it becomes independent. Here, what we have gone, we have gone for the antioxidant assays. 6 antioxidant assays we have conducted, that is DPPH, hydrogen peroxide, hydroxyl radical, superoxide, nitric oxide, and hypochlorous activity. Of this, all the 6 assays have given the very good result, and especially DPPH and the hydroxyl radical, as well as the this one, what do you say? Superoxide has given the wonderful result in comparison with their control. Then we have gone for the antimicrobial potential of the Ficus rootfield extract. We have used 2 sets of bacteria, 2 gram-positive, that is, we have the Staphylococcus aureus and we have the Bacillus subtilis, and the 2 gram-negative, that is E. coli and the Pseudomonas aeruginosa. In our study, we have seen that it was more effective for the gram-positive, maybe because of in the case of the gram-negative, there is a high lipid content and the low peptidoglycan content. Next slide. This is the result, like we have measured the zone of inhibition less than more than 10 millimeter we have considered for the good result, and you can see in the Staphylococcus aureus we have got the wonderful result. Next. This is the GCMS analysis of the ficus rumphi extract. We have used methodic extract of the ficus rumphi, where we have got around 30 compounds. We have outsourced to GNU. Yes. Then we have gone for the network pharmacology.
Last 1 minute you have.
How much?
Last 1 minute.
Okay. Oh, then you have to rush. Okay. We have gone for the reverse pharmacology studies. That is, it is the protein target prediction. I define the leads, then going to their network metabolites. This is one of the. Please skip this. Please. Yeah. These four compounds, that is lanostirol, beta-amidol, and hexadecanoic acid and stigmasterol, were promising in the anti-cancer studies. So first we have gone for the druggability. As we know, like when we go for molecular docking, the first thing what we do is the druggability of the phytocompounds analyzed through the Swiss ADME tool, where our compound were giving very good promising result. Then we have gone for the network pharmacology studies, where we have used the Swiss target prediction of the first 14 targets. We have went into the string database, where we have got in total 64 proteins, which is identified by these 4 phytocompounds. And out of this 64 proteins, 12 proteins were associated with the anti-cancer. So again, in that again, like we have focused on the renal cancer. So out of these 12 proteins, again the 9 proteins, it were again we have shortlisted up to the 9 renal cancer proteins. This is the net pharmacology network what we have got. That were with the yellow border, that is showing the renal carcinoma related target of the ficus rumphi. Yes, this is the sub-network, but only the 9 one is being focused. Next, in the molecular docking studies by the Autodoc tool, we have got good result with the beta-amidol and the lanostirol with the MDM2 gene with a score of minus 10.7 and minus 8.5 kilojoules. So this is outline, like where we could project the anti-cancerous property, where the ROS or the RNS, it leads to the stress in the environment, where normally the P53, the phytocompounds prevents the binding of the MDM2 with the P53, thereby arresting the cell cycle, and the uncontrolled proliferation is being. Yeah, this is the last part. Yeah. I'll just wind up. This is the cytotoxicity studies, which has shown the selectivity index of 3. This is the outcome, where we have identified the lanostirol and beta-amidol as the potential metabolites, and the renal cancer-associated target proteins identified by this network pharmacology studies. Inhibition of MDM2 P53 as well as the cyclin D1 can be a better therapeutic targets for the renal cancer treatment. The rest is the conventional assays, that is anti-proliferatory and the antioxidant studies and antimicrobial studies have a very positive result with this ficus rumphi. Thank you. Yeah.
If any question is there from the audience, if there is no question, we can move to the next talk from Anasuya Karzi. And please. Okay. And she's noting the time, so you have to keep the 7 minutes intact, and then 3 minutes you can ask the questions. If you take over 10 minutes, we will not go to ask questions, and we will not mark you for the question and answer. Okay? So be in 7 minutes. Okay?
So very good morning, everybody. Today I will be presenting my work based on isolation, screening, and detailed genomic analysis of the potential probiotic strain, that is Staphylococcus sp M005 isolate from Syrinus milky. So in recent years, that there has been a tremendous increase in the human population, and due to which there has been a pressure in the food supply sector. So in order to check upon this, various measures have been taken. By focusing upon the aquaculture area, which is the controlled cultivation of aquatic plants and animals, mainly focusing upon the pisciculture sector. Like pisciculture covers, like India itself is nearly 50% major producer of fishes, and it covers nearly 50% of the fish supply in the world. So in order to check upon like there for a continuous expansion of pisciculture, to check upon the epidemic diseases caused due to viruses, bacteria and all, commercial fish feeds needs to be given to them. So if you use commercial fish feeds, like the antibiotics are present in those commercial fish feeds, and it gives rise to resistant bacteria, which it produces subsequent contamination in the environment. So in order to check upon this, a new measure has been taken, that is the introduction of symbiotics in the fish feed. Symbiotics is composed mainly of two components, that is probiotics and prebiotics. As we all know, probiotics are dietary supplements containing potentially beneficial live bacteria, yeast or algae, and prebiotics are non-digestible feed components that improve the host intestinal balance. So to be considered as a probiotic, the live microorganisms should contain some characteristics, like particular characteristics, like they should not be easily available easily digestible in the digestive system, and they should be easily attached to the intestinal epithelium and colonize there. They should be non-pathogenic and non-toxic. Also, they should produce some antimicrobial substances in order to check upon the invasive pathogens. Similarly, prebiotics should contain these particular characteristics, like they should stimulate only selective microbiota in the gut, and also they should be able to resist against enzymatic digestion, and also short-chain fatty acids are produced after the degradation of prebiotics. So, symbiotics can be called as the safest and most effective solution for combating colonization and infection with enteric pathogens. So, to begin with, my work is to formulate a compound, symbiotic compound, based upon I have isolated a probiotic from the gut of Sirius nigella, that is a freshwater plant residing in the benthic zone, which consumes mainly the detritus. So I have tried to isolate a probiotic from the gut of Sirius nigella. So proper screening tests were done for antimicrobial assay against a common fish pathogen, that is the Aeromonas hydrophila, was done. And these are the various isolates that I have got from it. And these are the zone diameters. I have selected the MG05 as all the other bacteria belonging to that same zone diameter, producing the same zone diameter. They were physiologically same. Also, carbohydrate fermentation tests were done in order to check whether the bacteria could utilize various spectra of carbohydrates. So these are the out of 31 carbohydrates were able to be degraded by the isolated strain. And these are the antibiotic sensitivity tests that I have integrated by the UCAS guidelines. Erythromycins have become tetracycline resistant. In order to check whether my strain was hemolytic or not, it was found out that the strain MG05 was found out to be non-pathogenic. So phylogenetic characterization was done, and we can see that my isolated strain belonged to the cluster of Lactococcus lactis species, along with Lactococcus subspecies horni and Lactococcus lactis subspecies lactis. This is the whole genome phylogenetic analysis, and we can see that it belongs to, it is forming a separate branch in the cluster of Lactococcus lactis, and this you can see the Lactococcus xylosis has been changed into Lactococcus lactis recently. In order to check whether, I have studied the whole genome, and here I found out that potential genes involved in carbohydrate fermentation, like SGNH, SN hydrolase family protein, they could break down the ester, these are mainly SN hydrolase enzymes, and they can break down various cell wall compounds, like hemicellulose, cellulose, peptine, etc. These are potential genes involved in producing antimicrobial substances. These are these genes were activated under bile and acid stress. So now I have isolated a potentially probiotic strain, and now I am shifting to my prebiotic. I have tried to extract a prebiotic compound from waste product, horticultural waste product, that is the orange peels. I have compared it with the pectin, structure of pectin, and I found out by the pectin analysis that all the functional groups were present in the extracted material. I have formulated an MSM medium in which I have supplied the crude extract of my orange peel powder as the sole carboxes for the acylated strain, in order to find out whether the acylated strain could take up the carboxyls. So I have done a growth study. We can see that the acylated strain MG05 showed a tremendous increase in the first 12 hours with the carbon source that are provided. It can be seen that it is decreasing gradually up till 12th hour. So at the end, I would like to say that I have tried to isolate a potential probiotic with which I will try to formulate my extracted material from the plant source, and will formulate a symbiotic compound which can be fed to the fish in the aquaculture region. Thank you.
Now we are open for question answer. Any questions are there? No questions? So we can thank you, Anupriya, for the nice talk. So now we can move to the next presentation, which is from Yashmin Chaudhary. Please come and present your talk within 7 minutes.
So I'm going to present my work on phytochemical screening of our very own giloid, and also we have done some in silico work. So let us see what we have done. So these are the contents of our slide. Now, a lot of work has been done on very deadly diseases. Now, what is the definition of deadly diseases? People are working on cancer, respiratory diseases, cardiac diseases, but there is a lesser known disease which is still happening. It's ongoing. So reportedly, more than 700,000 people around the world have already developed antimicrobial resistance. And according to World Health Organization, if this continues, so by 2050, an estimated 10 million people will die every year due to diseases caused by bacteria which have developed resistance. And this is going to be very dangerous, much dangerous than diabetes, tuberculosis, and HIV combined. Therefore, a need for new plants or new medicinal plants may be the solution. So, Ginospora cardifolae is one such plant which has been known in Indian traditional medicine since ages. And there are so many benefits of using this plant which I have already written. So, our major goal was to screen out the phytochemicals and also to find out which was the best phytochemical or isolate which was responsible for the antimicrobial activity. Also, we have functionalized it using silver and gold nano and also performed the in silico studies. So this is the methodology that we have used, and the plant was collected from a kitchen garden from the Hindoguri Gaon of Soraido district, and it was also verified by the Government Survey of India. Then we used the solid extraction for extracting our extracts, and we used hexane, chloroform, methanol and aqueous solution, and as you can see that the highest yield was from the aqueous solvents followed by the methanol. Then we have also performed the preliminary phytochemical screening and using the standard protocols, and you can see from this slide that the highest yield was from the methanolic extract of the leaf, because the leaf and stem for both we have done it differently. Then we have also done the antioxidant activity using the DPPH radical scavenging, and we have used quercetin as our control. And this is towards your left, you can see for the percentage inhibition of all the extracts for stem, and the other side you can see for the leaf. And the take-home message from this slide would be that the stem methanolic extract showed the best antioxidant activity with IC50 values which were very comparable with the quercetin, our control. Then we have also performed the antimicrobial activity for this for the extracts, all the extracts, and we have for this we have considered three mostly bacteria. Those subtilis is there, E. coli and aeruginosa, and you can see that from this slide the take-home message was that the methanolic we used flash chromatography and tried to isolate the compounds. Then we performed the NMR and we found that this particular compound actually belongs to the cordifolicide family. Now the cordifolicide family has 5 compounds in its kitty. So we tried to nanofunctionalize it using silver and gold nanoparticles. So these are the SEM and ETX results of the gold nanoparticle, and as you can see, the spherical, these gold nanoparticles, they were forming very beautiful spherical particles, but they were not diffused onto the surface. Rather, when we performed the gold particle size analysis, we came to know that it was between 0.15 to 0.3 micrometer. And then we also performed the silver nanofunctionalization and the SEM and the EBIX results are here. You can see that for the silver nanoparticle, it was very nicely diffused. Nothing could be seen. Probably the particles were formed in nano size and they got diffused. That's why we couldn't find the particle size for this. We also did a comparative antimicrobial activity study for the isolate the silver nanoparticle as well as the gold nanoparticle, and we found that the silver nanoparticle showed excellent antimicrobial activity, and it was almost comparable to our control. We also performed the MIC and MBC studies, and this is for the aeruginosa, because aeruginosa was showing very good results. And also you can see that very less amount of silver nanoparticles, the gray part, was showing the activity. Then we tried to perform certain in silico studies. For this, we considered the tall V protein for aeruginosa. And what we did is, because there were 5 compounds, so we used the density functional theory to screen out which is the best compound. So according to the HOMO-LUMO gap, we found that the cordifolicide C was the best, most reactive compound. So we considered that compound and then we tried to dock it onto the tall V protein. Now this tall V protein of aeruginosa, it is present between, it's a part of the tall pulse system of aeruginosa and it's considered very, very important. Therefore, when we tried to see the binding, we found that it was very nicely binded in this protein, and we believe that this study would show light towards this problem of antimicrobial resistance, wherein silver nanoparticle will give a push so that we curb this problem. Thank you so much. These are the references that I used. Done by my student mostly. I'm sure it is a key important here. And also I'm thankful to Ankita, my artist, and my colleague, Dr. Subrat Sinha, for all the small talks we have, Dibrugarh University, for giving the opportunity and the lab facilities. And thank you so much.
Open for the question.
Yes. As you mentioned that the phytochemicals after the screening, the phytochemicals are used for to make the gold and silver nanoparticles, and then by the DFT calculation for company you used it. So did you select one this phytochemical, or from this one you make a scaffold of it?
No, actually it was the isolate that we took. Okay, the isolate was the cordifolicide family. The cordifolicide family has 5 compounds in its kitty. So when we actually nanofunctionalized it, we functionalized the isolate. So you cannot see which compound it is. Yes. And then later on for the in silico part, actually instead of doing for all 5, we did the density functional theory and choose one.
Based on the DFT calculation?
Yes, it's based on the DFT calculations, quantum calculations mostly.
No more questions. We can move to next talk, which is from Sridhara Kangli. So please come and present your talk.
So thank you everyone for listening.
Okay, thank you.
Speaking on development of an efficient bacterium for the potential bioremediation of recalcitrant antibiotic cyclopropysin from antibiotic pollutant cycle. So what happens is antibiotics, of course, they are a boon to human health care, but when we take antibiotics, they are discharged from our body intact without undergoing any sort of metabolic changes.
So these antibiotics, they get piled up in the environment and having a very low like molecular weight and high absorption, they are retained even to the wastewater treatment plant. So these piled up antibiotics, they are actually the micropollutants, encouraging the discharge of or the dissemination of antibiotic resistant bacteria and genes. So what happens is that certain tools like chemical and physical methodologies, they can be employed for removal of these antibiotics from the environment, but they are not very easily accessible in countries like us. So in India, the only method through which we can remove these pollutants from the environment is through bioremediation. And we have chosen ciprofloxacin, a second-gen fluoroquinolone antibiotic. The reason is the structure of ciprofloxacin having a very strong carbon-fluorine bond. This molecule is recalcitrant and hard to degrade through any methodologies. So we have chosen ciprofloxacin for the study. And keeping the view in mind of bioremediation, we have designed our study. That is, we collected some samples from an environment, antibiotic polluted environment. We enriched them and isolated stains which are putatively involved in degradation. So of the 3 stains that we found, 2 were negative and 1 were gram-positive stain. These are the few biochemical tests that we performed. We found that they could ferment a variety of sugars. This is necessary for us because bioremediation is greatly influenced if there is a co-metabolite present during the process. The list of antibiotic susceptibility profile of the isolates, and we have found that all the 3 strains are multi-drug resistant strains, and they are resistant to almost all the antibiotics that we have tested so far using distribution method. This table 3 draws our attention because we have checked the MIC of ciprofloxacin against all the 3 strains, and we found that MIC has gone up to 10 mg per ml, which is quite high because the amount of glucose that we supply for a bacteria to grow is around 5 to 8 milligram per ml. In that case, the MIC, that means it can resist up to 10 mg per ml. This gives us a thought that how is the resistance mechanism happening. So with this, we designed the next experiment, that is the growth study. We thought that maybe our strains are utilizing the antibiotic as a food, and instead of resisting, it is consuming the ciprofloxacin. So the 3 graphs above, they depict the growth study, and we've seen that all the 3 strains have increased by 3 log folds. And the graphs below, they show the antibacterial efficacy of the degraded products. That was also a concern because previous literature studies have shown that the degraded products are more toxic than the parent molecule. But we do not want the degraded products to be toxic. So we checked the antibacterial efficacy, and there has been a decrease in the zone diameter. Further, we went with the SG01, which was the most potential strain of the 3, and spectrophotometric quantification also shows that there has been a degradation in the ciprofloxacin concentration. And these are the corresponding graphs, that is the growth study and the antibacterial under the similar conditions. So on like all the experiments we did so far has been conducted in mineral salt media, where this antibiotic was used as the sole source of carbon and nitrogen. We haven't supplied any other source of carbon and nitrogen. This is the FTIR analysis to understand the structural changes that have taken place in the molecule, and we have seen a difference in the transmittance as well as certain bonds have been degraded and formed. The schematic representation of the genetic organization of SG01, it is a 5.5 mg genome and has a GC content of 57.12%. To understand the phylogenetic position, we went for a 16S RNA-based phylogenetic analysis, and Klebsiella comes out as a different branch from Quasivaricola. To further understand, we saw the whole genome-based phylogenetic analysis, and it shares a common ancestry with Klebsiella pneumoniae. Figure 8 is the genomic island. Why we wanted to see this is that because the genomic islands, they are the major ways through which all the antibiotic resistant genes are disseminated to the environment. And we have found in the genome of SG01, there are genes for multi-drug resistant efflux transporters, toxin antitoxin system, the type 6 secretion system for sharing the metabolites and transfer of macromolecules. And also the AA6, C6, which is the aminoglycoside acetyl transfer, is involved in acetylating ciprofloxacin molecule as well as giving resistance to this. This is also being found in the genomic island. Along with this, there are certain pathogenic factors and like genes related to adherence and cytophrone-mediated iron uptake, which we have observed in the genomic island of this strain. So this is the antibiotic resistant genes which have been plotted in the genome, and this is a multi-drug resistant strain, as we confirmed from the antibiotic susceptibility profile as well, and they have genes like for the resistance to beta-lactams, tetracyclines, polymyxins, quinolones, aminoglycosides, which are used to treat various infections. So we hypothesize a pathway for the degradation that it is not a single-step pathway, however, it's a cascade of reactions starting from N-acetylation, hydroxylation, desethylation, oxidation. And one of the main challenges that has been seen till date is that the degradation of the quinolone nucleus. However, our strain is as it can utilize this molecule as a food, which has been supported by both study and FTIR. We suggest that it can cleave the quinolone nucleus as well. The genes of the pathways have been listed, and there are a lot of gaps here, which we'll be doing, like filling up using certain omics technologies. And the list of genes which are involved in the degradation pathway belonging to various pathways like the protocyclic pathway and the homogeneous pathway. So in conclusion, we say that globally work has been done on the degradation of ciprofloxacin as well as other fluoroquinolones, but the degradation has ranged in terms of milligrams per liter. However, the strain that we have got, it can degrade in terms of grams per liter, which is huge as compared to work that has been done till date. And the second significance of the study is that the degraded products are not at all toxic as compared to the parent molecule that is being shown. So the list of few references, and thank you. So we have done it with the natural water, like we collected water from one of the rivers which is running through the main city. So because that is the one who gets the entire pollution dumped there. So we checked that. And we removed the other bacteria present there, we autoclaved it, and then we studied this.
Okay, no,
but
that
is
not
removing
natural
the,
environment.
Yeah, that is not the natural, but to develop the study with it, we did that. And this is also there in our program that we will be
Because
like,
you
using
are
it.
not
Application, yeah. Again, that application will do that.
Thank you very much. So next up we have from Dalal Emaya. So please, she can come and present the talk.
I'm Dalal Emaya. I will present part of my PhD work. It's talking about investigation to the root of folate and succinate in the regulation of mTOR-pathway-related genes and functional activation of NK cells. First, a brief introduction about NK cells. NK cell, we know that it is cytolytic phosphatolytic lymphocyte that has an important role in viral submission and also have anti-cancer capacity and antiviral capacity. So and also have important role in cooperating with the adaptive immune system. So any dysfunction or deficiency in NK cell has associated with cancer and viral infection. So brief, here we can see the importance of metabolism of NK cell like other immune cells. NK cell has very restrict reprogramming of their metabolism according to their state, which is during differentiation or during their functional activation. So during functional activation, for instance, we can see that there is NK cell will increase the glycolysis and the biomarker will enter the mitochondria, but it will not go through TCA cycle, but it will go instead for citrate malate shuttle, which is different from what we can see in usual TCA cycle. For that purpose also, mTOR pathway is very important in regulating many of oxidative phosphorylation metabolism and glycolysis and other metabolism in NK cell. So, which is important in controlling cell metabolism, proliferation, survival, and immunocell differentiation. That will be response for growth factors, hormones, and cytokines from the microenvironment. So, any decrease or increase in the nutrient, mainly amino acid, folate, and glucose, will affect the mTOR, which in result will affect the NK metabolism and the function. One of the most important nutrients we can see in the important in the sense of mTOR pathway regulation, we can see folate. Folate is vitamin B9 is very essential for regulating various intracellular cellular metabolic processes like energy production, ATP production, epigenetic control, and mainly methylation. We see here folate cycles. The byproduct from this folate cycle, it will be a main product for methylation cycle, also mitochondrial metabolism, and also protein synthesis and DNA and RNA synthesis, which is very important whether in any proliferating cells, if we are talking about immune cells or cancer cells. In tumor microenvironment, NK cell usually suppress more than other type of immunocells. That related to many things. One of them, we suggested that it will be metabolism. So reprogramming of cancer cell metabolism is also happening for their rapid proliferation and also adjusting with the new environment they have. So cancer cells will reprogram and their metabolism and use the majority and try to sequester most of the nutrient and oxygen, which create a hypoxic and acidic microenvironment, which also will suppress in the result the NK cell. So this suppressing will happen, this will be very important for cancer cells proliferation and survival. One of important, like TCA cycle is very important to create ATP molecules for energy. So in TCA cycle, one of the byproducts, we found succinate. Succinate is one of the products like lactate and fumarate, which is an oncommon metabolite, will suppress, after accumulation, will suppress the immunocells, mainly NK cells, which will help in the considered as an inhibitor for the immunosuppression, immunosuppressants. And succinate is considered as, for that, it's oncommetabolite considered as signaling molecule and considered in the key metabolic factor in cancer immunocytes. For that purpose, we aim to investigate what is the effect, because there's so few studies about what the importance of folate and the effect or impact of succinate accumulation in tumor microenvironment, which may have a role in suppressing NK cell. So we try to investigate the effect on some genes of mTOR pathway and some genes of functional activation of NK cell. For that purpose, we grown NK cell in folic acid-free RBMI media. We supplemented with folic acid or the other one of the other form, methyl tetrahydrofolic acid. That is usually used by the cells, which is like the active form of folate, for 72 hours. And we consider this concentration as a control supplement. And the other side also we did the same. Also we treated the cells with succinate with different concentration for 72 hours. And then we harvested the cells and analyzed some genes. Here we can see in folate pathway, DHFR, RFC, that is for reduced folate carrier one for the transporter, mTOR pathway, and NK cell functional gene, interferon gamma, berberine, and ganzanide. Here we can see the result for the effect of folate. We can see that folic acid over supplementation. We consider the 0.02 as a control because NK cell is suggested to grow in that concentration. So any increasing for 0.2 folic acid and folate deficiency also, we noticed that there's significant upregulation in the interferon gamma. But there was no that significant effect on the others. For mTOR pathway, also there is significant upregulation in mTOR and SREBP and HIF1 alpha during folate over supplementation and deficiency. For folate pathway, these main genes, we found that DHFR expression was upregulated in 0.2 folic acid supplementation. And downregulation in MTHFR gene when we see in the deficiency of folic acid, folic acid. For succinate, we found that succinate treatment will upregulate interferon gamma and granzyme B. And for mTOR pathway, HIF was upregulated in treatment with succinate, and mTOR pathway was downregulated. For folate pathway, we found that THFR expression was upregulated by succinate treatment. In conclusion, we can say that folate deficiency or over-supplementation and also succinate treatment could have a pseudo-hypoxic effect by significant aggregation of HIF1 alpha expression gene. SREBB gene expression also was upregulated in folic acid over-supplementation and downregulated in deficient condition. So, that have role in impairing glucose, glucose metabolism, which we saw that it is important for activation of impressor, and this is will affect their effective function. mTOR gene expression also was upregulated and in the condition of over-supplementation and downregulated in succinate treatment, which may also affect the NK cell proliferation and metabolic regulation. For folate deficiency and also folic acid high supplementation, succinate treatment will increase interferon gamma expression gene, and it's reported and already known that interferon gamma will have controversial effect in tumor microenvironment, dependent on the other circumstances from the effect of the other immunocells and how the metabolism is going on for cancer cells as well. In closing, we can suggest that our data that suggests that there is some regulatory effect or some regulatory role in folate and succinate on mTOR pathway-related gene and functional activation of NK cell. So altogether, folate concentration is very critical, and also succinate accumulation in tumor microenvironment could be one of the reasons that compromise the metabolism and effective function of NK cell. There's some references I used in the presentation. Thank you.
Audience side, please.
Are you planning to do the protein level?
Yeah, I'm doing that.
So thank you, Tula. So
Yes, sir.
next presentation from Jyoti Das, and she's from NIPGP from New Delhi. You can go ahead.
Today I will be talking about the importance of the alternative sigma factors RP N1 and N2 during bacterial microphagy. Before moving forward, let me introduce you to the term bacterial microphagy. So what is bacterial microphagy? Bacterial microphagy is nothing but when a bacteria utilize fungal mycelia for deriving its own nutrition. So we have isolated a microphagous bacteria called Burkholderia glandulae NGJ1 strain. This is the yellow color bacteria that we have isolated from healthy rice seed leaf. And here I am showing you a deadly necrotropic fungal pathogen called Rhizoctonia solani, which causes sheath blight disease in rice. So this is a typical microphagous interaction between this NGJ1 bacteria and the Rhizoctonia solani. Here you can see after 7 days of interaction, the yellow colored bacteria completely covered in the white color fungal mycelia. So this highlights that this can be important for controlling this deadly fungus. Also, when we pre-treat this fungal mycelia, sclerotia with the bacterial culture and then do an infection assay in rice, we can see the disease symptoms drastically reduced. So this brings us to the question whether we can use this bacteria for biocontrol of this fungal pathogen. But there are several concerns with the genus Burkholderia, as it is known to be a proliferative pathogen in many humans, animals and other plants. So instead of using the bacteria itself, we are interested to understand the molecular mechanism underlying this microphagous ability and use the genes or the gene products of this protein metabolites for the control of the fungal disease. So in this process, we have previously identified that the dietary secretion system, which is a protein secretion system used by various pathogenic bacteria to deliver effectors into the host cell. This dietary secretion system is important for the bacterial microphagy. So when we mutate one of the
component, it is HRCC, of this type 3 secretion system, we see that the mutant bacteria is not able to kill the fungal mycelium. So it highlights that the type 3 secretion system is also important for bacterial macrophages. Now that we have found some key components of bacterial macrophagy, we wanted to understand how it is regulated, how bacterial macrophagy is regulated in this NGG1 bacteria. So in bacteria, we know that for transcription to start, the sigma factor or the sigma subunit of the RNA polymerase does the recognition of the promoter site and then initiates the transcription. There are primary transcription factors which are constitutively expressed in the cell for general gene expression, and there are alternative sigma factors which are only specially expressed in the cell whenever there is any stress condition such as starvation or any growth in any different carbon source. So these alternative sigma factors, so sigma 54, which is about the student stock. So sigma 54 or the RPOS were first reported to be important for nitrogen assimilation in bacteria. That's why the name is RPOS. So some bacteria also encodes multiple copies of these RPOS in their genome. So considering their multifaceted role in bacterial physiology that has been previously reported in various pathogenic bacteria like Ralstonia solanacearum, Xanthomonas, these RPONs are important for the virulence, some motilities, and also for flagella biosynthesis in Xanthomonas. These RPONs are also important for free-living and symbiotic growth in Rhizobia. So considering their multifaceted role, we are interested to understand whether these have role in bacterial macrophagy or not. Now we have found that the bacteria, Mycobacterium bacteria NGG1, encodes 2 copies of this N1 and N2 in the genome, which has a core binding domain and a DNA binding domain. So when we did a qRT-PCR based expression analysis during microphagous interaction between the bacteria and the fungi, we found that in the bacteria, both these RP N1 and N2 are upregulated in the early phase of microphagous interaction. So this gives us a hint that this might be important for bacterial macrophages. To fully characterize the importance of these genes, we have created a single mutant of this RPN1 and N2 and a double mutant of where both these genes are mutated. So when we do an interaction study, we can see these mutants are not capable of causing the microphagous ability when in presence of the fungus, whereas the wild type is proficient in microphages. So this highlights that these genes are important for microphages. We also did some complementation studies where we complemented the full-length copy in these mutants and this could restore the microphagous ability. Now, an important attribute of bacterial macrophagy is that after interaction with the fungus, the bacteria, while utilizing its nutrients, the cell number also increases. So the bacterial biomass is increased. So with the wild type, after we count the bacteria, we can see in the wild type the bacterial cell number increases after interaction, whereas the mutants there is no cell growth. But in case of the complementation, again the number is increased. Now another important attribute of bacterial macrophagy is that after interaction with the bacteria, as I mentioned, bacterial cell number increases and the fungal cell death is induced. So to see fungal cell death, we did a confocal microscopic analysis using a propydium iodide dye, which is a light exclusion dye. Here you can see this Rhizophrenia solanacearum fungus. When it is leaving, this cell wall is intact and you can see the cell wall completely. But in case of interaction with the wild type bacteria, you can see the cell wall is degraded and hence you can see the nuclei of the fungus. But in case of the mutants, these are viable and hence you can see no nuclear staining. Complemented stain shows that this can again restore the fungal killing ability. So this highlights that these are important for bacterial macrophagy and induced cell death in fungus. Now the most important aspect of this study, whether these genes are important for biocontrollability of this bacteria or not. So we did an infection assay in rice and we can see here that when we treat these fungus with the wild type bacteria, the disease symptoms are drastically reduced, but in case of the mutants, the disease symptom is almost similar to that of control treated plants. This can also be coordinated with the quantification as a relative vascular sheath colonization, also by the pathogen. Also, when we did a triton blue staining, which shows that cell death is induced in the rice, you can see the wild type bacteria has very less cell death compared to the mutants and the mock treated plants.
So as I've already mentioned, that RPL targets promoter region and they recognizes the conservative DNA sequence. So using computational studies, we have identified that 17 of potential RPL targets that could be the targets of RPL. Now, as I have mentioned, that type 3 secretion system is important. We then check whether these set of genes have any type 3 secretion system signal or not. We found that 3 of them having potential T3 as a signal sequence. We did an expression analysis and found that some of the target genes are upregulated in the wild type but not in the mutant. You can see the potential T3 as effectors are also being. We also checked the expression of the type 3 apparatus encoding gene and found that it is upregulated in the wild type but not in the mutant states. So this study highlights that the mycophagous bacteria encodes 2 copies of RPOL in their genome, which are both important for biocontrol and its mycophagous activity. And we have also identified certain target genes of RPOL and which have potential T3 sequence. So this is a hypothetical model wherein in plant the bacteria, mycophagous bacteria is contact with several functions which can be endopathic and pathogen. So while sensing the presence of fungi, the bacteria might be upregulating its sigma factors like RPOS, which is important for T3 upregulation, and then it can deliver the effectors and mycophagy can happen. So this is I want to thank the organizers of this seminar for providing me the opportunity to present my work. This has been my topic of my PhD work, which I have carried under the guidance of Dr. Gopalji Jha, which is scientist at NIT, and I would like to thank DPT for funding.
Thank you, Jyoti. Please go ahead.
How did you create the matrix?
So it is insertion in plasmid integration by homologous recombination.
Last part of the profiling of the expression, you have done the protein estimation or only the heritage?
Related pieces, sir.
And the genome level when you did that is sequencing, genome sequencing. The mutative target finding?
We have already sequenced the genome, the dark genome, and from the we have used a PET scan where there is a software which can give the potential target. If you give the sequence motifs, and then it will show us like the potential target regions.
You are planning for the protein quantification?
Yes, sir. Like my juniors will be carrying forward this work.
So did you publish this first or no?
Sir, the part of this work is published in environmental microbiology.
Thank you, Jyoti. So next talk is from Nazrana Sultana. Please.
Virus, infectious pancreatic necrosis, this is virus. We know that the genetic code defines the sequence of amino acids in a polypeptide chain. And we know the standard genetic code that we have, it has 62 codons. We have one initiation codon that is AUG for methionine, and we have 3 termination codons, namely UAA, UGA, and UAG. Now, we know that codons have a property of degeneracy. That is, they use, for one amino acid, there can be more than one codons encoding the same amino acid. For example, if we take the example of this amino acid serine, it is encoded by 4 codons, that is UCU, UCC, UCA, UCG, and AGU and AGC. Now, the choice of any one codon for encoding this amino acid is sometimes non-uniform and biased. This introduces us to the term codon usage biasness. The non-uniform usage of synonymous codons is known as codon usage biasness, and this is species specific. That is, the trend of changes in the usage of codons is different in different organisms. And this is an important topic to be studied because any mutation in the synonymous codons can lead to diseases like Crohn's disease, endotropic sclerosis, and cystic fibrosis. Now, I need to study the codon usage biasness in infectious pancreatic necrosis virus, which has double-stranded RNA acid genetic material and it infects trout, salmon, and other fish species. The significance of this study, that is, what could this study lead us to? It would lead us to the nucleotide abundance in the concerned virus, its cup pattern and also the evolutionary forces involved in governing the cup pattern of this virus. These are just two of the papers which I have taken up from among all the papers I have reviewed for literature. First one speaks about the codon biases in Zika virus, and the second one speaks about the codon biases in tumor suppressor genes of human. My objectives are to determine the base contents, to study the pattern of usage of codons, to study the parity plot and also the neutrality plot for this virus. For materials and methodology, I have collected the sequences from NCBI. Then I have gone for parameter studies like compositional properties, ENC, RSCU, correspondence analysis, parity plot, and neutrality plot. After obtaining the results, then I have went for data interpretation. Now, the overall nucleotide content in this virus was observed in the CDS of this virus, and we found A to be the highest occurring, and followed by the nucleotide C, then by G, and then by T. We also found that the nucleotide content at the third codon position and found this one, that is C to be the most occurring. That is, we can say that for this virus, the codons which were most preferred were C and D. We also found out the overall GC and AT content and found GC to be more than the AT content. Not only in the overall case, but also for the codon position 3, we found that GC was more than AT. We found out the GC content at all the 3 codon positions and found it to be the highest at codon position 1, the second highest at codon position 3, and then the third highest at GC 2, that is the codon position 2. These are the codon usage trend, that is, we can see that all the codons, these are not unanimously used or like equally used. For example, this is least used, and there will be codons which are highly used, these ones. Now, with the help of RSU value, I found out the overrepresented and underrepresented codons. Underrepresented codons are the ones which will have RSU value less than 0.6. And the overrepresented codons are the ones which would be having RSU value more than 1.6. Now we found 15 underrepresented codons and 7 overrepresented codons. We also found the amino acid composition and found leucine to be highest occurring in this virus, virus. Now for the parity plot, I have taken 3 parity plots. 2-fold parity plot is for the amino acids with 2 codons, 4-fold for the amino acids with 4 synonymous codons, and similarly 6-fold for the amino acids with 6 synonymous codons. And since we found that these codons were appearing non-randomly, and hence this can say that the mutational pressure and natural selection affected the curve designing in this virus. For the correspondence analysis, we found random distribution of the AT and GCNP codons, which simplifies or implies that both natural selection and mutational pressure is influencing the curve designing in this virus. And the neutrality plot gave us a regression coefficient value of 0.1 to 0.9, which indicates a higher influence of natural selection in this virus than mutational pressure. So, we found that this virus CDS are GC rich, and nucleotide A occurred the most, and also at the third codon position nucleotide C occurred the most, that is, the codons which were most preferred were C and D ones, and we found an average ENC value of 47.90, which indicated low cup in this virus family. And the reason is that when it's an ENC value less than 35, we say that this is high cup, and when it's more than 35, we say it is low cup. We found 7 overrepresented and 15 underrepresented codons, and correspondence analysis showed both mutational pressure and natural selection to be affected in this virus, and regression coefficient value of GC3 and GC12 said that showed us that natural selection was the dominant force. So the conclusion, we found that all synonymous codons, that is 39, were not equally used. Mostly we found them to be varying between the range 0.6 to 1.6. And C and D codons were more preferred ones. And the neutrality plot showed us that natural selection was the dominant force in shaping the curve in this virus. I'm thankful to Assam University for providing all the necessary facilities, and I'm also thankful to CSIR for funding this project.
Thank you, Dr. So now we are open for question answer. So anybody have any questions they can ask, please.
So are you looking for like in this particular family of viruses, how they are?
Yes, sir. Actually, this is just a part of the family. This belongs to the family Virinavidae. And I have already published a paper regarding the Cotomyces viruses in the entire Virinavidae family. So this is just a part of it, just one virus.
And you have the host that it targets, which one?
Yes. Host is salmon and other fishes also, trouts.
Okay, so you have compared the same viruses in the host.
Yes, so we'll go for it. Like we'll go for the cup analysis in the host, and then we'll go for if they co-evolve or something.
Correct.
We'll study the host parasite relationship.
And essentially, to provide them with what conclusion that you are making, that will make sense when you will be actually doing the host part of it.
Yes, sure. Thank you.
Thank you, Nirjana. So next is from Ms. Saida Begum. Please come and present your talk in 7 minutes.
First of all, good afternoon everyone. Myself Shada Nisa. I am pursuing PhD from Vishwavati, West Bengal. Today I am presenting small portion of my research work. My presentation entitled with Unveiling the Phytochemical Profile, Biological Potential of Adenindra pavonina bark and in silico molecular docking study of its bioactive compound with the targeted protein. Now moving to the introductory portion. From ancient times we know that the natures are the sources of the medicines, and various medicines are obtained from the nature, and these natures are also used as a tribal medicinal system. Due to high demand of the medicines, scientists are paying attention to screen out the different pharmacological activities and isolate the phytochemical compound. So under this scenario, present work undertaken to investigate the phytochemical profiles, biological activity and in silico molecular docking of the bioactive compounds. Now, the main objective of my work is quantitative and qualitative screening of the methanolic extract of plant bark by HPLC and GCMS to determine the antioxidant, antimicrobial and anti-cancerous properties, and in silico molecular docking to investigate the antioxidant, antimicrobial and anti-cancerous activity of the pituitary bioactive compounds. Okay, now moving to the materials. Adenindra pavonina belongs to the leguminaceae plant. This plant is also known as red seed tree due to its seeds. Seeds is like a bead-like structure. And locally in Bengali it is known as Rakta Kambal because the bark inside it is red in color. Various etherological papers use this in treating various of the diseases such as dysentery, indigestion and bowels and etc. So the plant is collected from Shantiniketan, which is my workplace, and the drying and extraction is performed by the conventional method. Here we use the methanol as a maceration. Now further details, phytochemical screening, biological study and in silico molecular study is done by this plant extract following these methods. Now moving to the result section. First, phytochemical. In quantitative estimation of the phytochemical groups, we get that the plant contains significant amount of phenolics, flavonoid, tannin and alkaloid. Among these, we get maximum amount of phenolics. In HPLC, result shows that the plant contains 5 flavonoid and phenolic compounds which are shown in the chromatograph pics, and out of this higher amount of caffeic acid and synapic acid is present in the plant. In GCMS analysis, structures of the chemical compound which we get from the GCMS. And these are the chemical compounds which we get from the GCMS and along with their pharmacological activities which is reported by earlier scientists. Now moving to the biological properties, by DPPH and ABTS we determine the antioxidant property of the methanolic extract of the same bark. As we see in the result that IC50 value of the bark extract in both the plant extract is more or less same to the standard ascorbic acid. It depicts that it has antioxidant activities. Now, antimicrobial activity. Antimicrobial activity was carried out by other well diffusion method. Here we can take the four the bacteria, 2 gram positive and 2 gram negative. And here we can take the streptomycin as a positive control and DMSO as a negative control. We prepared the concentration of methanolic extract with using the DMSO for different concentration. And here we see that zone of inhibition, it shows good amount of zone of inhibition for 4 bacteria. It depicts that it has antimicrobial activity. Now the cytotoxicity activity was carried out by the NCT assay against 2 breast cancer cell lines MCF7 and MB2231 and the normal cell line which is 0 cell line. Here the result shows that the plant extract shows cell death for the 2 breast cancer cell line and it doesn't show any cytotoxic activity in case of normal cell line. And these are the photographs of the cell under the microscope that this shows cell death in various concentration for the breast cancer cell line and it doesn't show any cell morphological change for the normal cell. So it shows anti-cancerous activity against breast cancer cell line. So further in silico molecular docking was performed by using 23 phytochemical compounds which we get from GCMS with the 5 targeted protein of antioxidant, antimicrobial and anti-cancerous. Through IgM dock, we can screen the phytochemical compounds. Among the 23 phytochemical compounds, we get one compound, androsterone phenylmethyloxamine is the best compound for this. We can further detail study through IgM for orthodox 4.2 for this targeted protein in order to determine the relative strength and binding energy of ligand protein complex and analyzes the conformation origin of ligand binding site. These are the results of docking. From this investigation, we can conclude that this plant contains various phytochemical compounds which have various of pharmacological activities. So we should further investigate in details to get novel drug. That's all. Thank you.
Plan. Nangka kebaan kila bansha akasyid kila bansha hakanono ka climate kaba kidon. Nangka nisdangi kene kalong ban yarap kamtam yagito kibre kibomdan kam donjam to create and to promote a job opportunity, especially kamtam ikito self-help group. Ni approach ki self-help group kamtam hakito bisnong ki baydon ki self-help group ni legally ki badon ka potential ki badon kata ka jina aki ba ni ekren bati ni slot bati ni ai awareness program ni ai ka training ni ai na dosagam but dinata ni sa sampade ya imat sheki abila sam ni pe ki jingko endalabian ni buyback padeki ni pensyet ki kemudian badu tweet padeki hagador kababian katung ke jingan jauh tamarket nanta house mem alhajar katundai naso komen batas sorkar habagadon kani aroma mission Mensyuah kapor jogi, ausmen ahajar katsan, katru, katnyau, katra, ilamat jike bah. Ni perlu dalade, iki jenggot bawa ki expenditure, na dong ni perlu dalade. Ande yang ahajar katndai, kejiwan juga ni aroma mission. Kalau yang rap iki, kalau le ya skim converted skim, sigi blok kun ka NREGS skim, namta balle ka land development, balle yaki wedding, iki kita baru kaya rap ka skim. But usatli lani isi ni provide money ni ai yaki free sapling check ni farmer abang ni ai yaki ni jinthong kum kita jinthong ni bang lagi la mention ante different type of crop kum lemongrass citronella mentha peperita geranium vetiver winter green turmeric lavender kita ni jinthong kiba nila ai shiki nora bagian tu. Nama riba tamangi ni deki private. Damangi indon kajaka sibon. Ni elect shaki server group. Ni elect shaki itu ki private individual. Ki bagilah banle bantu ki jitung. Hadir bagi jitung kelabian. But nila banyak keren orang kasorka. Nama kadon ka scheme. Ban ai kum kaneka distillation unit. Lalu ke machine. Bantu lihat yang kini kiumpunya. Hendak kita lihat kiwan dipat sini ia kiumpunya. Kam market umdan je kuslai ETBS hadu benda mentah ke company kalau dia biasa soy kalau dia tebat ni hadu mentah ni lah ban penbiang ia jikwa joki. Jikwa wonton per month. Hadu mentah ni nang latang 300 and 400 liters a month. The company kan kam sibun ba di sibun kam. Ada hadu menenta ni parla ban ai yang kita cinta kam jaga company. Kata kita farm, this is our farm, this is our plantation. This is the self-help group women. They are doing with this plantation. Ye ihani idang le ye plantation dia pun koi ke le wedding di audit di shape pada ha kata distillation unit ini ki self group ki ba ki kentei ki pendankam ni ka bandrep ni ki jingthong. Next ini ki jingthong arjat ame ki nursery bagi le cutting nama kini geranium ni kam da bagi le cutting onde ba ni kam da ki seed ni kam da ki sapling. Sehingga kita balik cutting lagi, kita cutting apa polyhouse, ini bukan polyhouse, kita cutting lagi, and then we transplant to the field. Misalnya kita transplant di pair jomi, but kita distillation unit, kita distillation unit, ni warna itu dijitung, and then kita set ke machine. Kata machine ka capacity juga dengan machine dari 500 capacity. This distillation unit 500 capacity. 500 capacity ni yo kado ni tap yo jitu mangko pembahasan spa kilo of harvest 500 kg of harvest. We got oil around 3.5 or 4 liters oil. But it depends on the crop. Not same crop. Grass you can get 3.5, 3.9 per charge, but geranium we got only 500 kg of harvest. We got only 1 liter of geranium oil. But geranium oil is very expensive oil. Now market at the present for 14,000, not less than 14,000 per kg. Kita kiri keungkinian, this is the oil we've got from the crop. Ini kiri oil, ada nata nilai untuk ke packaging to sell outside the state. Kita kiri ke packaging, kabangi lebha ke packaging, we sell nangne ni pa support Calcutta, we sell to the Delhi, we sell to Lucknow, we sell to many states in India. Because this essential oil is very demand nowadays. Kita self-help group, kita contain women group in different block, different village. Kiwan hante apa berwa, I free training and then how to plan, how to cut. Paro ni le hante apa berwa, I ka training lagi ka bagian, and then ni sa I free supply to them. We contribute iki pemusafir hadena ta ade ni buyback iki ni tipat iki yang itu ki jengthong bat ni sateliat haden lan dak lihat kami kan dokong kenian ni siu iki dak pisa bat haden ta magi kila temen mah lagi ni sisi iki jinyo jongki kani additional income Tam-tam mai bagi traditional crop ka baju hiju thong hiju rap dre balai ni rap ni kini pada ngimju encourage ki tam-tam itu ki jaga ki bala dak don pa hiju thong kum ki traditional crop ni encourage ki tam-tam ki westland itu ki baju ishra untuk bat ni thong ni rap ka kum kita jaga bat manta especially hante apa giant joy itu ki mining area Hakaba kumno lapen sniu ini jaga ba unda de esu menta nile kumno menta daga bangito ini di baratland di westland kumno nile ketika di women group ni kai hande banthong iki menta yo ila de kile ko joy haki khamta mi di mining area kita ila jengam menta khamta itu ki ka soil kalau acidic kalau acid lah, dari mentah, lap, kalau tes indo, kalau normal, dah kami orang iya gini, arti pro, hagi teh jaga bang rap, napo Garo Hills, Gardner, napo Giant Hills, napo West Hills, napo Ribhoy, ibu pun kiwan iya kawan, jadi mentah, kamtam, hagi jingdon juga dengan aroma mission, kabar kala pushok mentah, apa state juga mereka. Alor jong kita kebangi hak di dunia sebat, ni jusen kumpulan sebat yang involve. Tinggi kuat pada bangsa ini, remaining, bagi kuat bantenek, alor di produk. Kibangi isna, bang isna makakum kita ki room refresher, isna ki kum kita aroma candle, isna kum kita ki so sabun, isna ki insect repellent, dan ki flomo, and then kantam you insect stick udon nanti insect stick. Ini even ni dah fit seed siapa aroma shell, berpenuh kami insect yang agar betul. Ngai tiba ini ki seed siapa biang, ini satu lagi bangin hitung, hagi di jaga di barang lain itu. Nang badak itu kisi, ini yo berpenuh kam, bansna yo insect stick. Kita tidak kami untuk isi cuba ada pun kuipa. Tinggi snai kita kami apa jelala jauh. Kita nampak sibun ban keren kami kakam kejumi kedebet hafil. Ngadang keren mende ngomba baru kami di don ki degree, don ki position di bagian kawan di biang ini. Jadi ngamu tahu ordinary person. Bang yang ini ban aji kelas itu. Yang hakmat joki di student kita ladang akhiran ada student hakmat joki kita isu lalong noika dengan kong noika kita isu kinora. Datang kita kam ke jurnal bang ini kai isu kinora. Banderet ia kita dijungkung. Kita ki sosio ekonomik tio iang te idon dibentar ke bayarap seki doi don dibentar ke bayar apa bagi chemical. Ini kijengthong ke bagi ni rep untuk kam fertilizer ini. Ini kijengthong kita bagi situng bet itu. Dan kam fertilizer, dan kam pesticide, dan kam insecticide. Baru ini kita long bet kijengthong ke pada lade kita long pada era. Kita jadi ke pada sekolah pada pergi yang kaba hamba Lada bisu bom sutu, kadang bom sutu. Sawan ini bom stone, kabang bom stone nanti. Sawan interact kan ya, sekali digikali, ngan jubah sekali, nik ege ba tu benkam, berkuah bankili. Ngam benkam sibun bankili hane orang ba bankili sibun ngatrai ba cikren kam tanjimut hani lemat ni ikren bet hafil demonstration, hafil habangi penisi kata kedai kamram kalesun kecil kamte agak kuat agak selak ndai dekam kecil kedai bet bantu bandai apa kilingkha kecil kata kedai kecil cabang semua bantu ai lingkot kumta success story kabang ayat limba kedai kam kaba yang ngatrai di kublai sibun kedai kapur ke lupa.
Marwin has got a stall there outside this community hall. So in case you want to interact with him, you can. But if there is one or two questions, maybe you can ask him now.
Maybe you have a lot of questions because here we see the only maybe any institute here regarding this aromatic and medicinal crop. Maybe you have a lot of questions. So if you can interact with me after this meeting, you go in my store and ask. I will answer the whole question.
All right. Thank you, Mr. Dashmi Marwan. I would like to thank
Next speaker. Yes. Okay. So that's Martin Majau. She is the proprietor of Smoky Falls unit, which is based in Ceylon. She founded the first coffee roaster in the Northeast India in the name of Smoky Falls Tribe Coffee. She completed her MSc in biochemistry from NEHU in 2014. She has received various awards like MIT Award by Government of Meghalaya, Vasundhara Award from Delhi, and Northeast Unsan Hero DSC Mumbai, etc. So now I will request her to present her speech. Thank you.
I'm Dr. Majau from Shillong. From Shillong, and also a former student of NEHU from biochemistry department, year 2004. And I have started a firm called Smoky Falls Tribe Coffee in the year 2008-2009. But the reason that led me to start the firm was due to my passion for my work, which is coffee. While I was doing my master's and went for my interview for my master's degree, I still remember the day when my professors asked me from which village do you come from, from which village. So I told the entire board that I belong to Terna village, and then they asked me, what is the special thing about the village? I told them we have coffee. So back then, that was in 2004, but I always have, my love for coffee has always been there, and I grew up having my own coffee with my late grandmother. So after I finished my MSc, I went back to my village and started my own firm, the Smoky Falls Tribe Coffee back then. And how I work is, when I first started, I started with my own coffee from the village, from Janna. Then slowly, I started procuring coffee from many farmers from Meghalaya. So here, we work with different farmers from the southern slopes of Cherrapunji, southern slopes of Peninsula, southern slopes of Tauki, and also from Garo Hills and Jainti Hills. So these are some of our farmers that we work with. And this is my first roaster. This was way back. Back then I was still working in SBI. I bought my first roaster, which is a 1 kg roaster, which I had actually tested my roasting. Prior to this, before buying my coffee roaster, I used to do pan roasting. So these are few of my packagings, coffee packets that we are selling in the state, in many retail shops, and also online. And these are our latest coffee packets. Our coffee now, we are selling a coffee in many cafes and many retail shops in the state, in the Northeast and also all over India. And also we have our regular customers from many countries around the world. We are shipping all India and we are shipping also globally. Besides the coffee, I've also ventured into Lakadong turmeric. We work with farmers from Lasken block, and we are working with few farmers which have high curcumin content Lakadong turmeric. And also we have, after I ventured into coffee, I've spent my time into coffee for many years. I've dedicated my, from 2009 till 2017-16, I've just worked with coffee. So I was like, slowly, working and growing my coffee. And after that, I ventured into food processing, fully, into a full food processing unit. So during the pandemic, we had actually to shut down a factory, and I was thinking what we should be making. During that time, people were having cold and cough, so we came up with this called a jelly. It's a spice jelly. It is made from Lakadong turmeric, which has a high curcumin content, along with black pepper, which comes from the southern slopes of Cherrapunji, ginger, lemon. So this is a very good product, which we made in our unit. And it has a very good amount of vitamins and and other things which is required for people who are having cold and cough. I'm not saying that this is a remedy for that, but it's helping. And thankfully now, when we started in the year 2020, when I started, it was very tough for me because to get the stability of the product was very, very difficult. To be able to have a stable jelly product, it was very, very difficult. I had to work over the years to learn many different packagings. So thankfully, I think so because of what I learned back then during my BSc with Sir Joshi and also MSc from biochemistry, that also helped me in using, few of the things that I've learned to incorporate into making this product. And thankfully now I have a stable product and we can sell and ship them all India. So at least when we get back to the customers and inquire about the stability, now it has a stable shelf life. See, coming from the Northeast, you've seen products, especially when you go to different fairs and different stalls. Stability of a product is something which we, the Northeast, especially from Meghalaya, we always struggle. So maybe sometimes shelf life is just around 60 to 90 days, not more than that. So that has been a problem. So over the years, with practice and experience, we could come up with a stable shelf life. And so this product also we're selling it, in many retail stores in Shillong and also outside Shillong. And then later, after venturing to coffee and also into Lakadong, then I was introduced to jackfruit. Meghalaya is home to jackfruit, and every year, 400 lakh ton of jackfruit goes wasted in Meghalaya, especially in Garo Hills and also southern slopes of Dauki and Cherrapunji. So I was asked to make some products, of course, from jackfruit. I've also come up with a few jackfruit drinks and jackfruit flour and jackfruit other products. But one of the products which stands out from what we have been working is Jack Tikki. It is like an Indian snack. I believe you've heard of aloo tikki. So this is Jack Tikki, which is 100% green jackfruit and has and also with no added coloring. So the color comes from the spices that we've added. Again, this is where we had been trying to push this into the market. We have been sending to Kerala, to Karnataka, to a group of people who have been working with jackfruit. We have an all India group. So we've been sending. So what happened is that over the months, since 3 years ago now, it's been 3 years for me now working with this product. To get the shelf life and stability was very, very difficult again. One thing is packaging. Second thing is the stability of the product in itself. Again, slowly, with time, with experiencing, and also with having a base which you can hold the product. Thankfully now this product is a gluten-free and it's 100% pure vegetarian. So it's and it doesn't have garlic, onion, ginger as well. So we have gone wholly fully into spices and blends. So this is also gluten-free, and it's a very good product that will go into the market. We also have a good demand from Kerala and from Karnataka, and I hope so we'll be able to produce this by next year, like, you know, for now we are going slowly into a few retail stores. This is the product which is known as Jack-Tiki. Then now also from Smoky Falls we are also going into first, I think so many of you have never heard, when I first came out, people from the Northeast have never heard of coffee. So we introduced Meghalaya coffee to the world. Now it's also introducing Meghalaya chocolate. I don't think so many of you have heard of Meghalaya growing cocoa pots, growing cocoa plant. So I've just ventured and traveled across Meghalaya and worked with different farmers, and we have a cocoa plant. And actually the cocoa plant has been growing for the past 6-7 years, and this 6-7 years later, this is the first fruit. This year was the first fruit that we got, and we are getting to work with the cocoa plant. And we have done after harvesting, and then we have gone into fermentation and roasting. So in a few months' time, we'll be able to produce chocolate from our own state, which will be Meghalaya's first chocolate. We always, like everywhere, like people when they make chocolate by buying the cocoa powder from outside the state. Now I believe from this year onward, because by the end of this month, we'll be getting around 1.5 to 2 lakh pots of cocoa pots, not the beans but the cocoa pots, the yellow and the red one. So we hope so to be able to go into good production on this. So we are also selling online. After so much, like making products and everything, we've also gone online. We have our own website called Meghalaya's Finest. At first, we have made a website called Smoky Falls Tribe Coffee, but the website didn't work due to some technical issues. Where we are now live with our own website called Meghalaya's Finest, where we'll be able to put on our own finest products from Meghalaya. And also we are also available online in Amazon as well, and shipping all India. So these are the few of the things that I'm doing. And I believe I work with Bestily, from Aroma Mission, where we also do the packaging for aromatic oils, where we source from his farm, a wintergreen, peppermint, geranium, and turmeric, and we supply them to Meghalaya village. And also I run a resort called Smoky Falls Resort. It is in a place where we grow our coffee and where my coffee is started. So this is the place where we grow our coffee and the neighboring places. So these are the few things that I could tell you, and I believe that I'm really sorry for being late today as well. Thank you so much, sir, and thank you to the entire department for inviting and giving us an opportunity to talk. And I believe that the students here, should get encouraged to go well in making biofertilizers. They are selling all over the country and outside the countries. There are 2 women from biotechnology background. They have their own tissue culture lab in Tezpur and also in Manipur. They have started. We also invited them to come, but I think so they were not available this time. Hopefully maybe on the next we can invite them, because they are also doing very well in the field of entrepreneurship, being from biochemistry and biotechnology background. So I being from biochemistry background, it really helped me a lot, in understanding the product, understanding your own work that you're doing. It really helped. Because here we don't have training. In other parts of the country, one of the best things is that entrepreneurs, they get to visit labs, they get to visit food labs, and they get to experiment, their own work. Here we don't have. So being from this background, even all of you, I believe you really have much more what you can do, like supposing, a rural entrepreneur. He will be able to make a product. I've been working with many other rural entrepreneurs, being a resource person, and visit many other rural entrepreneurs. The one thing is that they have good products. Our rural people have good products. But the thing is that the moment we turn them into products, the moment we package them, they can never sustain more than a month because of certain issues like package value addition to the product. So being from this background, I believe that you can always look, and now since the government has many schemes, I being a beneficiary from PMHP, as a youth you can go into the government portal, PMHP, and take out schemes and loans, which I have myself taken a loan. Call the PMHP to buy the first coffee roaster. And also now, since our Department of Science and Technology, they also have many schemes for young entrepreneurs who are willing to open a lab, like even a normal lab for, medical lab or maybe other food lab. They have many other schemes, in tissue culture also. Like someone asked, there is also a scheme for starting a tissue culture unit as well. So I believe these are all a few suggestions that I want to encourage students from the university to also, apart from looking applying for jobs, you also have can also start from now to understand what you are good at and also your passion is, and you can also develop that and go into entrepreneurship and you'll be able to get a job. I myself now I'm giving employment to 82 direct employees and also work with around 400 to 600 farmers across Meghalaya. So this is one thing that which you can do and reach out with entrepreneurship. Thank you so much once again.
For this business, and I will request the participants, those who have any query, they can discuss with her in our bioentrepreneurs startups stalls. Thank you so much, madam. Yes. So the last speaker is Sri Amit Pridarshan, who is the CEO of Kalsi, an entrepreneur who has been leading a multidisciplinary tech team with the mission to solve various industry challenges. He is also the recipient of Best Startup Awardee at India LCD. So I will request him please start to speak.
Good afternoon, everyone. When I was planning my initial first set of talk, I thought I'll thank Nehu and the department and panelists for being here. But first I'll thank you all for staying till the end of the session while the lunch is waiting behind. So thank you so much. As the title of the slide goes, from Northeast to Middle East. So we all know we are in Northeast. Middle East is the place that exports oil to all over the world, crude oil. And we are into that, we are into petroleum biotechnology. So when I stand and speak here, I must recognize the efforts of hundreds of people who have worked with me in past 5 years, who may or may not have continued to work with me, but they were very instrumental in this startup journey. I will not go into explaining much of the technology, but I would rather say it cannot be done alone. You need a very good team if you want to. You can start with one person, but you need a very good team. So having said that, let me give you a brief background on my journey and my team's journey on entrepreneurship. So the first line says that we are the largest petroleum biotechnology startup in the country, and that is, by the way, true, not a marketing gimmick. Then we are a team of chemical engineers, biotechnologists, statisticians. We are a multidisciplinary R&D team. Our primary work area is industrial biotechnology. We ventured into industrial AI also for quite some time because of certain reasons that I'll explain. We have a collaboration with IIT Guwahati and 23 other institutes across the globe, including some of them being in Europe and USA as well as in University of California and other places. So I will not name all the IITs and other places, naming IIT Guwahati because currently we are there. One of our offices is there, and they were the first to help us out. About our team, they are far more qualified than I am. They are better in every field, so I have already explained that. Our expertise is that we have worked extensively with extreme environment microbes, particularly those coming out of oil wells. I'll come to the story of oil in a little bit, because oil, Northeast is a very significant player in the India's oil story. But we really are not aware of it, so I will go into that a little bit later. We have patented a system to produce 15,000 liters of microbes at a oil well site, which can be an isolated place where no electricity, even food is available. So it is in such an isolated environment we have built a capacity to do it. And everything started right here in Shillong. And to be more precise, most of the things started right here in Rishi's lab. So, see, world's second oil well was drilled in Northeast. It was drilled in Assam, Upper Assam, called Dikpur. In South Asia's first oil well was drilled in Northeast. That was the first time neither Russia nor any other country, particularly in Asian region, none of them have had this. So oil was first drilled in, and today also Northeast supplies 40% of oil to our entire country. But we do not really know about it. So we are a startup into that sector, and we have built technologies using biotechnology to cater to that sector, and that's how we have moved ahead. You know, it is most useful commodity. Why it is useful? Because for past 60 years, at least 60 wars have been fought on oil. Any war you take, the major reason is oil. So it is an important commodity. Crude oil is produced from wells that are drilled into the surface of the earth, which can go up to 6 kilometers deep. So there is a hole dug from the surface to the subsurface that can be a 6 kilometer long drill borewell kind of thing. And there is an impurity that comes along with it, which is sand. And in India, there are around 18,000 wells. 70% of those wells produce sand. And there are close to 0 companies in India that provide sand influx control services, and none of them but through biotechnology means. So this is the market we are talking about. The existing methods were there, they had their own problems, and of course we have outcompeted them in their problems. That's why we are in the picture here. So our idea was very simple: find out certain microorganisms that have the capacity to bind the sand grains from where, so that it does not come up. So basically we are making bricks out of sand that is coming up, and if we control the amount of nutrition, we can control the metabolic activity and a lot of other, like on a larger scale. So that was the idea. It seemed all great, but there were some problems here. The technical challenges were these. The temperature at the place where we are planning to inject our microorganisms would have at least 80 to 90 degrees Celsius, can go up to 150. So you all are microbiologists. You can tell me at 90 degrees Celsius temperature how difficult it is for a microbe to work and give you the results that you taken in lab. Very high pressures, fluctuating pH, and salinity up to 50,000 ppm, and these are the numbers that you generally do not encounter in the lab unless you are specially working for extreme environment. And apart from all this, then everything has to happen precisely at 3 kilometers or 6 kilometers, whatever the depth is, at that depth, and it has to work precisely, because if it does not work, you have just ruined a 100 crore national property, because oil will cost you almost that much. So risks are involved, that's why players are less. It's not that we are very innovative or anything, we just were not very aware when we started. But once we had taken up the challenge, we had no other option, because we have invested a lot of things into it. And yes, for this particular method that I am talking about, injecting microorganisms in a well to consolidate sand or produce cement, whatever, is nowhere done in the world till now. We started 5 years ago. There are no established protocols, no guidelines, no reference papers to look into, and there is absolutely nothing except for the idea that you have in the mind. So if I am talking about my journey of entrepreneurship, it is all about luck, because there is an even bigger challenge out here that we have to encompass. We had no background in biosciences, all of us. So everything started based on biosciences, all idea was created on that. It needed not only biosciences, it needed deep understanding of biosciences, because we were pushing the boundary of the entire field in itself. But 2-3 of us who started, we had no clue about biosciences, we were all petroleum engineers. So that's when luck had to play its role. We were looking all around Shillong, and I was studying in Shillong. We were looking all around, all the places, all the professors. We literally went to all the professors. If you are talking about any institute you name, I went to all the professors, asking, can you help me build this? And there were problems, not only technical, but yeah, bureaucratical problems can also be there. Then one fine day, I showed up in front of Joshi sir on a Monday morning without appointment, without knowing, without even, I had not even seen his face, and I explained the technology to him saying, sir, we want to do it, and sir said, we will not take the burden of teaching you microbiology, rather I will help you out with something else. So that's how it started. He gave us one of his brightest students who was very instrumental in our initial days, running the experiment, designing the experiments. From there our journey started. So if you ask me why we were doing this, what was the drive? I was studying petroleum engineering, I was in third year of my undergrad that time when we started. What was the drive? So definitely drive was not any intellectual thing that I got or any vision that I had, because I had never worked anywhere else before. The only drive I had was, if I do not do this, somebody else is going to do it in some time. And I'll be seeing this and it will not be a good show to watch actually. So that was the only drive, to be honest, and I was, all of us we were looking for everything that we could have. We even did not have a clue how to go ahead, but somehow things panned out. So this is a very good quote from Swami Vivekananda. If you read this, I will not explain this. This is a very simple thing that he says. Take up one idea and pursue it unless you do it. There is no ifs and buts about it. And I am not encouraging you to do it as an entrepreneur, because a lot of things are at stake when you are an entrepreneur. A lot of investors' money is involved, a lot of your money and time and your team's time and resources are involved. But if you are like me in a college, you have nothing to worry except for failure. So you can do this, and that's why I think you are the right audience for this kind of my lecture on luck. So because if I do it in an investor summit, nothing is going to happen. So the early experiments, as I said, in Professor Joshi's lab, I do not have the photos of those experiments, but it was done in small petri plates, borrowed from Joshi's lab, with chemicals borrowed from Joshi's lab. And I'll tell you one more interesting thing. Crude oil is a national property. It's not that you can go out and take crude oil from anywhere because you want to run experiments. You need to take permission of a body called Central Industrial Security Force, even if you are working in an oil industry, and then they'll allow you to take maybe 100 ml of oil. We found somebody whose father was in a refinery and he snuck out 100 ml of crude oil for us from a pit, because you cannot take it from the main line, and that's how, sir, the crude oil that I showed you initially, that was from there. If you are trying to consolidate sand, another important factor apart from microbes and crude oil is sand, obviously, and that has to come from the surface of like 5 kilometers, 6 kilometers surface of that. So that was another mountain to climb, but we climbed that somehow. Again, we had to validate the theory, like the technical aspects of it. These are the logistical challenges that anybody has to overcome. Theory was also not vetted, and we had to simulate conditions of an oil well in lab, so that we can even go to an oil company and say that this works, because they will not give you 100 crore well just to experiment your stuff because you seem to be a bright student, which by the way is still in third year of college, so not bright at all. So these are the early experiments. We ran a lot of experiments. I will not go into those details, but thousands and thousands of experiments were run in past 4 or 5 years. And out of those experiments that we have run, we have filed for 13 patents in last 4 years. So that is the kind of work that we have done, starting out of Northeast, starting out of Joshi's lab, even without a background. In the beginning of a startup, there are many things at stake, and I can tell you my story, what was at stake. I had like zero relevant credentials to start a business. Nobody would give me even 1,000 rupees saying, hey, you will make it into 1,500 rupees and give me tomorrow. There was no business credentials, there was no technology credentials, so we were literally going nowhere. We had no credentials to start. So if somebody says you that you should start in your own domain, much better. But I am you can say an outlier, a lucky outlier to be precise. The things can work this way also, so don't be worried about if you do not fall into the regular picture of a great entrepreneur. Because I was not, and currently I am, sorry. Placements. In 4th year you have placements, have paid huge fees, and companies come to your college, they'll give you a job. And I know what I had to do and in fact I was not looking even to become an entrepreneur. That's the wild part of it. I was looking for a lab wherein I could validate this idea so that I can give it over to some oil companies while I am in college. So that was the entire, I was looking for a lab, and that's why I wrote to hundreds of people. Even when we came to Joshi sir, he said we want to experiment, because we had no idea what a startup is like. So there is a risk of placements, losing jobs. Fourth year is an important time in your college final year. You have to be focused, you have to be preparing for interviews. I was, after my college hours, I used to come to Joshi sir's lab, sometimes on Saturdays and Sundays as well, in night, and with his help, we used to work on the technology after attending my class. Mind you, I did not leave any class because that was also hanging. Knock every door, some opened, like we suddenly got an email from somebody in ONGC and Oil India saying, hey, look, we cannot give you labs, but if you, what you are asking for is half an hour time to discuss your idea that we can give you, you can come over. So it was all nice and fine, but they called me over to Duliajan, which is near Dibrugarh. So I had to travel all the way from Shillong to Dibrugarh with making sure that I can attend my classes and all those things, have a meeting, and then come back and do all those things. So that was the thing. In Shillong, we were living in a PG. We had no address even to register a company that is required. And now the regulations have changed. Back in 2017, when I was doing all this, regulations were different. They were far tedious to go ahead with that. And in all this procedure, I did not ask money from my parents, simply because I did not want it to be shut down before we even started. So all this money was taken from my friends who used to travel with me, and it was not a lot of money at that point, looking from this point in time, but that was a great helper. That's why I said hundreds of people have, because of helped me. I wrote to many people. That meeting that I was talking about in Duliajan, somehow people got interested in that because apparently it was one of their biggest problems. If you look at the geology of wells in the northeast and read about the sand problem, all the north, 70% of India's well produce sand at one point or the other, whichever is in sandstone. So they said this looks like an interesting idea, but we cannot give you lab to work because labs are not allowed for you. There is no policy. So what you can do is we have come up with a startup fund. You can apply to that. If you are selected, we might fund you to start your lab. And that's where, and they said, we applied for that. And out of around, there were many companies, 120 companies that were there. 2 of us were selected, one was us, and we were still in college that time. Still, I had to go to class after being selected also. So, everything was going great at that point in time, but there was one other problem that we had to figure out. If they select us really, are we going to leave our college? Or are we going to say that although you are giving us money, we cannot take it? And a third problem popped up saying, look, you are doing great, but we cannot give money to you, right? We need some entity to pay money to, we are a PSU. So we must make a company now. And that's where the startup story started. So it was a very long journey of almost 1 year before we even started at that moment. For 8 months, I was just meeting Oil India's people, all the big shots of the industry, right? All directors and head of the departments of this thing. And it was not happening in Shillong, it was happening in Duliajan, Guwahati, and Delhi. And I was traveling during my college and Joshi sir's lab to all these places as well. Convinced board of directors of Oil India Limited in a board meeting of while in college. So everything was happening in college. Had to convince head of finance with my financial proposal, which I had zero idea of. So I'll tell you how I did it. This is all great, but I'll tell you how I did it. In order to do one thing, I used to be very diligent, and I used to be awake 18 hours a day probably sometimes, just to make one presentation or cover one topic. And for this financial model I'm talking about, I read 2 full books in 4 days just to get an idea of how to do it even at a basic level. So all of this was being done, and 8 months of scrutiny, MOU, Oil India signed an MOU with us, and IIT Guwahati became a partner at the same time, and they paid us around 2 crore rupees, and we were still in college. So now I had just, luckily just after 1 month of this, things panned out, we passed out of college, and I had no other option but to do this. The later experiments happened at IIT Guwahati. Joshi sir used to come to IIT Guwahati multiple times during this period. We used to, even Professor Tandon, who was here yesterday, saw us in our early days, he remembers us till now. We started from scratch, and then we had experimental backup, but we did not have any equipment as such. With all those things we did, and the goal was simple, it has to go into an oil well. I really did not care whether we write any papers, any patents or anything, it has to go into an oil well and it has to validate itself because that was the mission. Every work that was done in that direction, that it has to go into oil well and no pseudo success validation that you can get in the beginning that okay, sand is consolidating and we are good to go. No. After all this handling, a lot of things are there in between. We went to the field trials and we did successful field trials in a well in a place called Abjan. It took 7 days. We injected 15,000 liters of microorganisms. Things panned out as expected. We got a letter from Oil India saying that this technology works, and then we were on our runway. And then after that, we did a few other trials. The last trial that I did was done in the month of March 2020. So as soon as we did our second well, 2 days later, entire city was locked down. In fact, one of my teammates was locked down at a guest house in Upper Assam without, like, he was eating bread and omelette every day for 40 days. So that kind of situation he was stuck in. As Swami Vivekananda says, the history of the world is the history of women who had faith in themselves. So in this period, what we did is we assembled a team, we went into industrial biotechnology and we really hit it. Now we have a major portion of our revenue coming from industrial biotechnology, industrial AI as well. So that also we did because we had only access to computers. Now, where are we today? Apart from all the story, where are we today? All India's major oil companies are partners. Indian Oil, ONGC, Oil India Limited, Vedanta. In little while we'll have Reliance also. So these are the major companies in India are our partners. Few of them are our investors as well. We have become the largest petroleum biotechnology startup because one thing, there are not many, and the second thing is that it is a wild idea to do so. We have global presence of our technology. We are supplying our technology to Nigeria, to France, to Middle East as in Saudi Arabia, UAE, and all those places. We have several agreements with them. We are doing it in Indonesia and okay. So we collaborated with ISRO last month to do some microbiological experiments with them, and after 3 years of painstaking scrutiny, there was that also. Multiple projects, and we currently have multiple projects in the pipeline. We are a regular invitee of the world's largest petroleum exhibition. We have some distributor network there as well now. ISRO, we signed an MOU as I told you. We got the best startup award at India Energy Week that was concluded like 20 days ago. So it was done in Bangalore and it was a pan-India event and it was a very big event in the petroleum industry. So these are our few achievements. We are known for our categories, national and international. So I have already explained you my pain, so the achievements come out of that. And this is a very nice quote that you can just note it down if you do not have time. The man in the arena. It is not the critique who counts. It is the man who actually stands in the arena. It counts whether you fail or succeed. You have to take that shot. Otherwise, there is no point. So I will end my talk here. This is my number if you want to connect. This is my email ID. Thank you so much for your time, and thank you, Joshi sir, for all the support that you have given me.
Shamil Sridarsan. So in the very challenging days, now you are emerging an excellent entrepreneur in your field. Really your journey may be a learning lesson for many young startups. Thank you very much.
In case if there's any questions, maybe one question in case if you want.
Yes.
In case if there's any question. All right. So if there's no question, then maybe do.
What species of microbes?
We started with Bacillus pasteurii, but we have taken it into a complete different level. We have a big list of microbes that we have currently for the
Do we still have that microbe that is starting?
Yes, you can take it from Joshi sir. Any other questions?
Just one question.
Yes, sir.
Can it be useful for oil spills also?
We do not work in oil spills, sir. But I am sure there must have been experiments done with Bacillus pasteurii for oil spills. But we are not into that yet. But there are many microbes that work with oil spills. There is a big company. Delhi does a lot of things with oil spills. Yes, ma'am. Thank you. You have been a great audience.
National Seminar on Emerging Trends in Biological Science, a Northeast India Perspective, organized by the Department of Biotechnology and Bioinformatics, NEBU, and in collaboration with BRDC and BIONES by the Incubator Facility NEBU, Tura. I would like to express our gratitude to our 5 speakers, Dr. Sinha, Dr. A. Chaliha, Mr. B. Marwen, Ms. Majal, and Sri Amit Priyadashini for your excellent talk on your journey as an entrepreneur. And moreover, it has been a motivation for our young scholars how you have started your work from as a student, as an entrepreneur, and what we have seen today, your great achievements, all right? So, and how you have contributed for the role in the growth of the economy of this part of Northeast region. Thank you so much, and in case we have any more discussion or questions, we will do it during after lunch when we have the panel discussion. Thank you.
Thank you, Professor. Now we would like to conclude that session with the felicitation program. For that, I would like to invite Professor S.R. Joshi to kindly felicitate Professor R.N.K. Huru. I would like to call upon Professor Joshi again to felicitate Dr. B.K. Mishra. Sinha up on stage. Thank you, ma'am. Next, I would like to call upon Dr. A.K. Chaliha. Marvin, to please come up on stage. Thank you, sir. Now I request Srimati Dasumardan Majal to please come up on stage. Thank you, ma'am. Thank you, sir. Now with that, we have come to the end of this session. We will...
I think I must place on record on behalf of the organizing committee and the department our sincere gratitude to Dr. Sinha, Dr. Chalia, Bob Esther, Ms. Dasu, and Abhi. It's so pleasing when you see your youngsters. Over the past 35 years, we have handled Professor Chatterjee, who is my senior colleague this year. I think probably for 40 years we have been in the classroom. But when you see, these offshoots of our exercise that we do, blooming in life, that is the greatest satisfaction a teacher can get. This is where I'm so elated to see Ms. Dasu doing so well, Amit becoming global figure, Ms. Dasu already on that platform, and these youngsters, Dr. Sinha, Dr. Charlie, or Vesta, you are all half my age doing so well. You are motivated by yourself, and I see the youngsters take up. It need not be in your platter and off your menu, but you can still make it. Thank you very much. We break now, and we'll assemble around 2:15 for the next session. Thank you. I must thank also our chairpersons. We have Professor R.N.K. Huru, our elder sister, and Professor B.K. Mishra from Athura campus, who himself is looking after the biomass. incubator facility at our university. Thank you, Madam and Sir, for your time. Thank you.
For today's discussion comprised of Professor S.R. Rao. We just finished with one session and we just started with lunch. We'll wait until sir finishes. But the rest of us can come up to the dais and kindly take your seat. We have Dr. S.K. Nandi, if you are around. Dr. Nandi ji. Okay. We have Dr. Preeti Singh, who is, I think, having her lunch. She will be joining. I'm so sorry that we are intervening between the lunch. We have Dr. Swapnil Sinha. Madam, kindly come up to the dais and take a place. We have Sri Abhijit Saikia. We have Mr. Abhijit Saikia is here. Then we have Ms. Dasumarlin Mazao. Ms. Please come up. And Sri Vestarli Marwin. Marwin, who was here. And we have Sri W.J. Rani, Sri T. Enjo, and I'll be moderating the session. So we have two of us here, and the rest of the panelists. Can I request Dr. Nandi, kindly come up, and we have Sri Abhijit Saikia, Vesterli, Barani, I think I can see Barani, please come up, and Yanju, to kindly come up to the dais and kindly take your seat. We are going to start with the discussion. Right. And can you come back? Beginning, doesn't matter. We'll wait, sir, don't worry. Rao sir will wait. It's not in a hurry. You can have your meal, take your time, don't worry. We have the panel of experts that, on the dais. We have Ms. Dasu Marlin Mazao. We have Dr. S.K. Nandi. Then we have Professor S.R. Rao, my colleague, who is superintendent. We have Dr. Preeti Singh, who just joined. We have Dr. Swapnil Sinha. We have W.J. Rani with us. We have Thomas. I call him as Thomas. That is the advantage of being a little older in age. So Thomas Yanjo. So we begin. We have Professor Danton, who is here with us, my teacher. And Professor Chatterjee, my senior colleague from the department, and all our distinguished participants who are here. So the panel discussion that we have is themed as from academia to startups, opportunities and challenges in Northeast region. How we are going to go about is, I'll just be having some questions that I have in my mind thrown to the panelists, and then we start interacting on that. And as the interaction begins, we may also invite some of the queries from the participant, but let's also make it a point that we will not be stretching this beyond 45 minutes. But I know as the discussion goes and the panelists start responding to the queries that I begin, that's how we incite the entire discussion. You will also have a lot of queries. So we'll have to restrict at some point of time. So let me take my seat and then begin. I'm not going in terms of severity or any orders. I just made some questions, looking at the panelists. I would like to start with Dr. Preeti Singh. There are some of us who are all in the field. Professor Rao happens to be a pure academician who has done a lot of translational work. So from research lab to the field, he has a lot of experience too. So I will start with Dr. Preeti Singh. The question that I thought when we planned this is, what potential do you see in Northeast, if somebody wants to start a company, do you see a potential in, if that be the case, what would be the areas?
Thank you for giving me this privilege to answer this question and just express myself. Maybe I'm not right in all the aspects, but I have interacted with a lot of young researchers here in last 2 days, and I found that all the young minds, scientific minds from Northeast deserve more. And we, as in India, we are youngest population in the world, but where do we represent in the scientific community? Our representation is not proper. So I don't believe in grabbing low-hanging fruits. I am a little ambitious person, and I believe that Northeast has a lot of biodiversity that need to be scientifically proven. And for this reason, we need intense collaborations with the rest of India, not to concise and work with the strength rather than the weakness. If you try to do everything yourself, I think it's not a good idea. And if we collaborate extensively and prove our beliefs through the scientific experimentation and not to grab just low-hanging fruits like 1 year, 2 year, begin a startup and try to make money or something, but go for hardcore R&D where you can absorb your scientific minds. They are ambitious. They need more, and I believe in them. Thank you.
Thank you, Dr. Singh. Yeah, the point that we understand is, as scientists, we do a lot of work in the lab. I think there is collaboration that we also believe in, but something that needs also to be done as validation. That's what is the point. You cannot do everything on your own. You got to look out for resources where you can get the help chipped in. That's the point, Simit. And next is, I have a question for Dr. Swapnil Sinha. How did you basically initiate this idea that you can have a company? Because we have a lot of us here. What triggers that particular idea that I can have a company, and what made you to think so?
I was answering that question while my presentation. Truly speaking, I was bored of 9-to-5 job. That was the reason. And second was I wanted to have some creative independence. Those were the two main reasons, and I think that was and it was always like us when I was sitting with, because my company technology is DNA aptamers, so we were sitting with this aptamers, which I have made for bacteria, and I was thinking, what will happen? I will just publish one paper, and I will go for a patent, get it, and then what next? So that next part was something I wanted to do something, it should go out. And being in IIT, I have seen there are huge number of patents, over 500 patents sitting there. But I think 0.001% of patents, they actually see the actual market. They don't get translated. Because of this reason, because people, they don't have that drive that, instead of completing their degree, they should take it forward. And the claims that you have made in the paper that this could be a game changer in, say, diagnostics or healthcare, you really have to do it, and you cannot do it until you put it to commercialization. That translation needs to happen. So that was the main drive, if you ask me, to opening a company and moving from actually workbench to market.
Yeah, as I see from academics, when we talked about patents 3 years ago, there was a craze, and there was a drag to get a lot of patent files and we were interested in getting our promotion through the patent, which happened, which still happens. But we are not looking at translational aspect of this patent, which we are very rightly saying that, when we try to patent something, it must have something that can reach out to the society. You are very well taken. Dr. Sinha, just to continue with you saying that, the first thing is your interest, very importantly. What feasibility that you would see in Northeast, if one has to start in terms of the resources that we have? What do you think immediately somebody can think of starting a startup?
A very good question. I think resources we have in terms That's not where Northeast lands. What is required here is to build a very strong startup ecosystem and a mindset, which I believe is a little far away from, when you talk about the startup ecosystem in Bangalore or in Delhi or even in Kolkata. So that system, that ecosystem mindset needs to be built among the youth. And things are changing because now, see, previously there was a problem on land. People don't, investors especially, I'll tell you a recent example. We did a investor meet at IIT Guwahati, which was organized by Bioinvest on 14th and 15th February. We had a hard time convincing 20 plus investors to come to the campus and sit with the startups, be in the pitching session. Dr. Nandi was there, he was in the pre-meeting. But still, we were able to bring in 25 India's best investment houses. They came because they were invited by IIT Guwahati, rather than coming all the way to Guwahati, traveling and meeting the startup ecosystem. Though their outlook changed when they were taking their flight back home. So that is a good point. So we need to build that ecosystem, and I was talking to Phil a while ago before the panel discussion started, that this ecosystem, that needs to be built, in which youth needs to be, exposed to what things are available to you in terms of incubator network, in terms of connects, mentorship networks. So all these things need to be exposed to the youth. They should know. So that is the thing that is lacking here, what I see.
Thank you, madam. Now, as far as the students are concerned, and my experience as a teacher, our students have this little complex. Don't take me wrong. That probably I'll not be able to compete with the brains that are present in the mainland. I'll give you one instance of our students where All India University has started this scheme of innovation where the ideas is basically ideation, and they submit their ideas and we send them for All India level competition. Last year we had 3 of them selected from the university, but due to certain personal problem, only 2 of them, one of them could attend that. And he came with the second prize, All India. So this is what, it's that we don't come forward. Very well taken. And we have Dr. Nandi would be there. Dr. Nandi, this question is, how difficult it is to start a company in terms of funding in Northeast? Because a lot of us, as we saw, our young entrepreneurs, they have knocked, every door, but then they could somehow manage. So I would, just to highlight what is the nature of funding that one can explore in terms of starting a startup in Northeast?
You were asking about the startup funding, startup funding. The startup funding is available, or what amount of money? I am not sure.
See, suppose I want to start a small company, for example. I want to do something. I may be needing, let's say I need 3 lakhs of rupees in my mind just to set up a room and a small gadget, as Dr. Dasu started with the grinder, for example. So where do I go to approach? I may not have that money. How do I manage to get that money?
There are different kind of funds which will perhaps facilitated by different innovators. Say if you are looking for a traditional business or a business which is using the traditional lookups, then you can go for the banking schemes and etc. But all the banking schemes has after a certain period you have to give it back. But if your business or the ideas you are looking forward, you are taking forward are somehow innovative, then there are different kind of grants from different people. Say from state government agency, if we are going to say the grant is here, they are giving the kickstart grant of 5 lakhs, which is totally free. That is, if your project is good and it is selected by them, they will give a kickstart grant of that. Apart from that, from FIDAC and others, and from us, Barnes people, there are grants of prototype building grants. That is when you are thinking that is your idea is ready and it can be supported by the existing facts that you can make the prototype, then you can apply for certain funds like biotechnology ignition grant, and even there is a biotechnology ignition grant called Future entirely for Northeastern region, big NER, that gives a hefty amount of 50 lakhs. So we all the biologists, there are 7 biologists people in the Northeast India who all will facilitate you to write or draft your proposal and how to make that protocol, how to we can provide and we can facilitate him in having that. Apart from that, there are also many funds, such as if we are talking, we are deciding in the Northeastern University Academic Institute. I believe there will be a lot of research that has been done which are innovative and which are already in tier 3. As in laboratory scale, you have demonstrated that is the models are working. Then for all those funds, all those projects, we can avail the funds from CIBRI. This is another fund from BIAC which is comparable to to big that Bangladesh Education Grant, it will be 50 lakhs from the BIAC, and the rest of the fund you really want that the 50% of the rest of the fund will be provided by BIAC. So this is totally available only for the academicians, the you people serve. So if you have any kind of idea for that also, you can have one. You just only to have another industrial partner addition to that who will do the industrial application. So there are a lot of scope regarding to funding. If you are individual, then you can go for PMEGP, which will be perhaps the easiest one to take. And if your product is innovative, then there are multiple grants, not the loans, which you can avail, and you can look into us, or you can just move to us to facilitate you in availing those grants and throughout the journey. That is why you are making the product. Then also there will be some help from us regarding mentorship and infrastructure.
Thank you, Dr. Nandi. Just to add to that, as elaborately put by Dr. Nandi, see, if you are interested, if you have some idea in mind, every institution has the incubator facility, we call it as bio incubator, where they incubate you and then they also mentor you. We have bio-based in Tura, they are doing, even in Shillong we have incubator in Neho. Every institution is supposed to have that. Guwahati IIT is doing wonders there. So you can approach them, they will incubate you, and then they will also tell you how to go about getting the finances, if I can add to what you were saying. We have Ms. Rasuma.
And one thing, there is the best thing about this facilitation is that you can look into your nearest partners and you can work at your own place also if you have that facility at your place. So the facilitation can be done from remotely also if you have all those facilities in your place. So that is the very big plus, that you do not need to relocate. Because if you are in Nehu, Shillong, you can walk in here and the nearest partners can facilitate you online.
Thank you. I think this is for the youngsters to take up it, as we have the students there. Now I'll come to Ms. Dasumali Mazal. You have done your MSc in biochemistry. We had two situations here. We had Amin and his group who were basically petroleum engineers and they jumped into microbes. I remember the first time when they saw that microbes, so what is this? And you had a biochemistry background which came very, very handy in at least the food products that you're doing. Now, how do you relate the kind of education that we have? Should we only choose the area that are confident and trained in, or one can also jump out of that arena if I have the idea? And how much did your education help you to grow up as an entrepreneur?
Yes, as a student of biochemistry, it helped me a lot in my entrepreneurship journey, especially in food processing. It has really helped in so many fields, like especially when developing a product. It takes months and sometimes it takes years and understanding. And one of the main factors, like application of what we have learned during our degrees and master's degrees. It really help us in understanding the kind of product that we put into the market and the kind of what is needed to make the product stable. So we have to see, especially when you make a product which has a high acidic level, to make it, to stabilize it, what are the ingredients you have to give, understanding it and using certain, parameters and work with certain instruments. These are things which really help me a lot as a biochemistry student. So, like few products that we have developed, right from jackfruit to spice jellies, these are very, very important, which has a very basic chemistry base, a biochemistry base that we need. Working because like, we can make any other product, fine, but then again understanding what the product has, especially now people are more into lifestyle and healthy lifestyle and having healthy food. So understanding that in each of the product that we put in different parts, different components of food, the nutrition is very important. So having like in the products coming from giant food, which is high nutritious, understanding it and giving it to the people, giving it to the consumer that they need this high in vitamin A, high in vitamin B. We have these products which are very good. So we have a group of people who take a product because of certain basic nutrition needs as well. And especially like jellies, we have our own group of customers which they take it as a daily basis because they feel that it helps them a lot in the immune system. So these are very important things that we understand, and applying my knowledge from my biochemistry background into developing these products is very important. And I believe there are few also like some of the science students and biochemistry students as well. They also really really help them and especially into developing a product.
Thank you. Yeah. But then also that if someone has a background or particular subject of knowledge, it should not stop you from entering into any other area where you want to read and learn and then you begin. I think that's, thank you so much for your comment. I'll come to Barani. He was what do you call it, the tinkling force when we thought of having this bioentrepreneurship exhibition. Thank you, Barani, for all that motivation and the guidance that you provided us initially. I'm told that you provide training to bioentrepreneurs, who they have certain ideas, they can approach you and then get some kind of training or how to go about proposal making or thinking to start up. Now, do you consider ACEs as a factor when we take these people in, or the background is a factor, or the education is a factor, or what is the modus operandi in terms of enrolling people or guiding them through your resources?
Thank you, Professor Joshi. Yes, with regards to trainings, initially what we do is we as a kind of an incubator, state incubator. What we actually do is initially what we have started is we are concentrating only in incubation and pre-incubation for getting all the startups or entrepreneurs to support them, to take them from one level to another level. But due to the requirement that we see, after a couple of interactions, and we saw that some of the entrepreneurs, I'm just telling this as in general, irrespective of bio-entrepreneurs or something, but we, as Madam Brady was mentioning, the biodiversity that is here in India. So we have a lot of entrepreneurs who are into that sector as well. So what we did was we have tied up our collaboration with Media State Skill Development Society, who are our training, they provide us training providers for entrepreneurs. So we have recently sent three batch of entrepreneurs to Calcutta and Bhopal for training in packaging. Training in packaging and training in bamboo and handicraft. So these are advanced training into that. So whenever this requirement, we connect with various experts, department, or those who are in depth with, who knows deep about those kind of support. So we provide that kind of support to the entrepreneurs so that they can upskill themselves and know what they need to do. So in that part, that is what we do, and we are working closely with let's say BRDC, Biogas program, and even engaging a lot of academics in the process. And these are the supports that we try to provide to the entrepreneurs. And when it comes to how can we as a incubator supports anyone who works in. So we also have like Dr. Mandi was mentioning the financial support that we give. We also provide them with all these things, but it should have some kind of innovative or innovation-based kind of idea. So, irrespective of that, we are also providing them with so many kind of support when it comes to legal aspect as well. So suppose now for an entrepreneur who comes to our hub and wants to do something, they don't know how to register a company or prepare a project proposal, or just simple things as filing a GST or registering for GST. So we facilitate that to some of our empanel partners who can help them in that. And proposal, yes, of course, anyone can come in and just tell us what the idea is all about, and we kind of see the feasibility there and give them what is, how they can go ahead with it. So yeah, we accept all this, we provide the support.
Thank you, Barani. So you heard that if you want upscaling yourself, the skills are available to provide that support. Initially, also there is a support. And just to add to that, we have Bionest Mathura campus, which is doing wonderful work. We have Dr. Nandi. There are also training programs that are offered to PG and PhD students, right? And is there any charge that you take for providing training to such bio incubation that you have? Is there any initial charge or admission fee that you charge for them?
Charge actually depends on what kind of training it is. Generally, if it is skill development training for the rural entrepreneurs, we never charge. For the students, we never charge. But if it is for some advanced instrumentation or something like that, and we need to invite some company and etc. from outside, sometimes it was charged. But generally, these are very nominal, so the charges are not will be a problem. And even if anybody needs any kind of from us, they just want to. So the work can be made free. That is not a problem at all.
So that should not be hindrance. So anybody wanting training can also approach Bionest to Dr. Nandi is there always 24/7 available. And we can also provide you the details and contact numbers. Next is, Bart Thomas, who is here. He's from BRDC Bioresource Development Center, Government of Meghalaya. Bart Thomas, you do organize a lot of programs and workshops. Do you also have anything related to the startups or a person wanting to do something very, very specific from the background that they come from? Are these workshops target oriented, or you organize very general kind of workshops which are more academics?
Thank you very much, Professor Joshi. When we look at first of all, I would like to just give a background on this center. The center is a research center in which we have certain infrastructures. So these infrastructures comprises of tissue culture lab, then we have biofertilizer lab, then beside that we are also trying to promote sustainable green agriculture, since we are the organic certifying agency industrial. Then besides this, we are doing some other activities which we have civil infrastructure, like we have this polyhouse, we have this seed conservation unit, then we also have this cold storage for conservation of all these processed food or any other product that is required. So what this center is, for the last few years, we have been trying to offer these infrastructures to aspiring entrepreneurs. Because when we look at most of the entrepreneurs, when they require infrastructure to set up their infrastructure, say for example, when we look at enterprises in tissue culture, if they need such facilities, it will be a very Herculean task for them to set up the tissue culture lab. And for them to utilize, to ask for funding to set up the tissue culture lab, then they will waste most of their time in setting up the lab and then trying out different types of experiments to complete what they are aspiring for. So we as a center, we are trying to offer all these infrastructure to be kind of incubation unit for these aspiring entrepreneurs. Say for example, when we look at the biofertilizer lab, in which we are producing all these microbial bionicotinoids. So this biofertilizer lab that we have, it can be utilized by those entrepreneurs who are willing to set up enterprises in production of biofertilizers. So they can come and establish, they will come and do their experiment, try all their experiments there to produce their desired products, because we as a government, we can afford to take that risk. We can afford, we are able to invest on the consumables, on all the aspects, even the manpower, we can invest for a certain amount of years until they have reached that stage in which they can stand on their own. So these are some of the offers that the center is trying to give. Rather than, we are not expert into training, we are not expert into marketing and all, but we are offering this infrastructure for the entrepreneurs.
Thank you, Thomas. Now we come to the academician. We have lots around. My question is, we are all bounded by 4 walls, and what we teach in the classroom, maybe I was just talking, we have experts who are here, and the kind of research that we did, and with the publication that we have hundreds in number, and when I look at it, probably we would have had 30 to 40 patents from whatever we have done. We did not know how to go about it. We are learning it now. The question that I have is, now what do you think the education system needs to be doing just to change this perception? The present education system, of course, NEP has a lot of aspects in this, so that more people will be or will get encouraged to get or venture into this aspects of entrepreneurship or starting a startup. What changes the education should bring now?
A very pertinent point that you have raised, Professor Joshi. We are all fellow teachers, we are here, and our learned senior teachers are also with us. Yes, we have to identify the reason what academicians' role in the present today scenario, where, especially for biotechnologies, the interface with the startups and thereby progression of the region as per the need of the places. That has to be discussed and decided. Today, we have both the components. Academicians, you have kept on one side, and then we have very prospective youngsters who will really take up the startups and then make them successful for future technological innovations. To find an actual answer, I tell you, we should compare where startups have been great success and what academicians have been doing there, and what we have missed, perhaps what if we have missed the point, what is that point? That is to be corrected. Then, I'll tell you, academicians have a lot of ideas. I tell you so many ideas that enough number of startups cannot translate those ideas into this. I tell you as an academician, out of my 33 years experience. But that's not happening. There are no, I'm talking about the Northeast India, and I will focus on Meghalaya and this part of the region. Why? Actually, I found an answer after July when I superannuated and I happened to be in Bangalore and Andhra Pradesh. Actually, the startup environment was actually developed and it was promoted by the industries, because they are the technology stakeholders. So they have a lot of things and they want certain technologies to be developed. I will tell you one example. I happened to be in my village in the last 6 months, first time after so many years. So there was one industrialist who came how to harvest coconuts without actually you can climb the tree and get them. It was I asked the industrialist, I happened to be there, why you need it? He says that nowadays nobody wants to climb the tree and harvest the coconut. You give us a machine, let it go, let it bring down. And you will be wondered. There is one English university in Vijayawada, Vignan University. They have already given him a machine. So now that started, how it has come? Because there is a need. And that need was, of course, was given by an industrialist, but the society is benefited. You know, I always tell you, during my childhood, when we used to harvest the coconut, the person who used to climb the tree, many times they used to have an accidental fall, or they used to have the bites by scorpions and other insects. Now there is nothing like that. So a need has been fulfilled. Now an academician can help the technical person how to build up that machine. Now simply you have to remotely operate. And in fact I was offered to, we own a little bit land, these are there. So I was offered that, I may purchase one. So what I am saying is that there is a big role for industries to come to the academicians and to tell them what they want. For example, if the region wants more plantation of bamboo to be done to afforestation, technologies are available within the NIBU, within the plant biotechnology people working in the university. And if they want some biochemical agent, for example, packing material, suppose they want to develop from the pilot materials, there is a technology or there is a knowledge available, but that cannot be translated unless there is some industrial involvement is also there. Only academicians and enthusiastic startups can. So that ETBS should also be brought into the picture. We should request government to please encourage the industrialization in the meaningful industrialization for sustainable development. That's all.
Thank you.
Thank you.
I think Dr. Sina has a point to make. Yeah.
I would just like to add something. This is an excellent point made by Dr. Rao, and you started your monologue with this, that have you missed anything? No, I think academicians never miss anything teaching their students, and professors like you don't miss anything. What I believe, I was a teacher once. I'm a daughter of a teacher, so I understand this. Professors always give best, more than they can to the students, so you don't miss anything. What I feel is like the way you said that there is a huge gap between academia and industry, and that needs to be filled. Like you said that the need, so that identification of pain point, that is done by the innovators, like me, maybe like she's there. So that is done by the innovators, that they understand, they identify the need, but then they need to connect with the right industry person to tell them that, okay, fine, you are addressing this need, but is this address of the need or the solution can bring money after, once you have developed the product. So, building a product is the task of the innovator with the education that you have provided, which is, pie, that is, there is no, crosses there, it is there already. What needs to be done is to tell an innovator that yes, because ultimately it comes to money. You have to bring that product to the market, to the right people, invest the money at the right time when it is needed, so that it has a huge outreach. That sustainability and that is a gap which is existing between basically academia and industry. So that gap needs to be filled by what I would say, their strategies need to be designed, which will help a budding entrepreneur tell them to solve that problem which has a very good market sustainability. So that needs to be done, I guess. And another thing I would like to just, what Dr. Nandi pointed out, that in the beginning, when you are starting your startup journey, there is no dearth of funds. What happens, what I have looking at the startup since 2-3 years from now, I mean, I'm sitting across the table with startup in front of me. Where they struggle, I tell you. It's not just about the Northeast startups, it's all over. They, in the beginning, to develop the POC or the prototype, they have ample grants. Government grants, they have not angel investors, they don't come at that point, but they have huge government support in which they can develop the POC or prototype. What they struggle is when they are ready with the prototype, when they are doing limited market validation, and when they are making this commercialization process. There they need to raise investments by angel funds or VCs. There they fail. I have seen many startups fail because they think that half of their journey through the startup, they rely on grant money. They don't really come out of government grant and look for, because when you talk to investors, they are brutal. They are brutal. And it's given because they are investing crores of money on your enterprise, they are risking it based on the technology whose science they don't understand. So they only look at the market dynamics.
Thank you, Dr. Sinha, for that input. We would love to listen from the experts because they are there. We can have a long day discussion. But we'll invite some questions from our audience. Are there some queries that is already there, fiddling in your brain? So, yeah, there is one. Can we provide him the mic, please? He's one of our research scholars from the Department of Biotechnology and Bioinformatics. Yeah, Rick.
Sorry, sir, am I audible from here, sir? Yes. Sir, first of all, it's very nice to know so many things.
Can you come up here?
Yes.
Okay.
So first of all, sir, it's very it's so many new things we can do as a student, as a scholar. As a PhD degree is just a degree, it's not giving you a guarantee to a job. And looking at the current scenario about jobs, I think I'm from West Bengal, it's very difficult to get a government job right now. So a lot of entrepreneurship opportunities are there, as we came to know. I already passed my post-graduation and now I'm doing my PhD. But sir, it's just an interest, I want to know. After becoming an entrepreneur, many of you have seen so many entrepreneurs. How is it necessary to, all the finishing schools that the Western countries they have, right from the graduation or master stage, they teach the student how to become an entrepreneur. It's a part of the curriculum as a finishing school. Before they finish the school, they have a prototype or they know the skill, social skill and the marketing skill, and if we don't have to go for a separate MBA to learn all those things. Do you think that it is necessary to become a part of the curriculum that we learn all those things?
To cut short, I think what he's asking is, should entrepreneurship be a part of the curriculum itself from school level? I think,
I don't think so, because see, entrepreneurship, there are lots to do in your life. Many people want to become something else, they want to be a doctor, they want to be, entrepreneurship is just one thing that you want to be in your life. And like everything, you have skill schools. And believe me, I think many of who are entrepreneurs here, she is there, Amit is there, might be somewhere. So it's not something that you acquire through books. I'm telling you a hard fact. When you really become an entrepreneur is when you fail terribly. I have one failed startup experience, and that experience actually made me a successful entrepreneur. So, of course, there are a lot of entrepreneurship development programs that you undergo while you have to be innovator first. Entrepreneur is something that comes after. You have to be a creative thinker first. You know what you want to do, that innovation part should be first. Once you are through with that, then you need to learn the skill set. So entrepreneurship is basically a skill set. Innovation is, by birth, maybe you are innovative. So, and for that we have, when we enter, innovators in our program, and when we board them in our cohorts and all, we have a full-fledged entrepreneurship development program. We bring in a lot of mentors. There is no, and of course there are a lot of books on it. If you do an MBA, there is a lot of strategic management, all those things will teach you how to be an entrepreneur. But separate course, mandatory, I don't take, I should agree with Dr. Rao, I don't know what your views on it. So I don't think so there should be a separate mandatory course for entrepreneurship in schools. Could be a subset.
Thank you, Dr. Sinha. See, we are also running short of time. Our guest of honor and chief guest are already waiting. We may take two more questions. One is Pritham. The tabla wala, he plays beautiful tabla. Actually, we had a wonderful evening yesterday. So let's move from tabla to Anupram. Come.
Good afternoon, respected so my question is like, suppose when one is going to start a startup, sell in the market. For this, the idea of the quality of the product is important, or for this, the need of the product in the market is more important.
So you're talking about asking, is the quality of the product important for selling it? Yes.
Sir, it is important, but like I want to let you know, like suppose one make a product, but the product quality is good, but the need of the product in the market it is more important, or if the quality is good, then it can be.
Yeah.
So this question yourself, when you go to the market, right, if say you have one product there is one brand Colgate, right, and there's another some toothpaste, which will you buy? You buy Colgate, right? So building a brand is very important. We have an entire separate, core program which tells you how to build your brand. And you only build your brand when you have a full quality checks. And I can agree with she will agree with me. So quality is both things are important. See, the need is the first thing, that is the drive which gets you started at the first place. So you have started your journey. So that is finished. That part is already over because you have already addressed and you started a product building. But then putting that product in the right quality so that users will come back. You can make a trust build up among your customers. That is the foremost thing being an entrepreneur, being a businessman for that.
Sir, I am a master's student. How can we become an entrepreneur? Can you explain how can the journey, the failures we going to face?
How failures can make you success? That's what the madam said,
I think he is trying to ask, hello everyone. So we actually presented our innovation yesterday, and I think he is asking that as a master student, so from innovation to entrepreneur, what are the hurdles that he will face, and who will guide you, like those kind of things?
Okay.
Yes, ma'am. Yes.
This question is very close to my heart because I am working in a startup where we daily basis, momentary basis, we face this challenge. The problem is we want to develop R&D based platform, and of course we are in India, so we have to think about monetary terms as well. So it's like the point where you want to compromise on, that totally depends on you, and you have to be innovative. How will you sustain your business? And 2 points I would like to make here, that I do not know, when I talk to young generation people, they are mixed up with industry and the business. Industry is different, business is different, to my understanding. Second thing is, nobody needs to tell you what is entrepreneur. There is an entrepreneur deep in our hearts. It is just like you identify the need, that something is needed. It need not to be a product. Uber is not a product. Ola is not a product. Amazon is not a product. What they are doing, they are building a business. Business means they identify that there are customers, there are retailers, they want to sell, and they need a platform. What we identified from my own experience, we talked to so many students all across Mumbai, South. They wanted to try their hands on. They wanted to do their own experiments, and there was no platform available. When we were running out of money, we got ample amount of students, with the money in their pocket, they wanted to validate what they were taught in the theory. And we had money in our hands. Of course, we got philanthropy support as well. I'm thankful to them. But I'm just saying that so we are not in industry in real terms. Our challenge is we want to build a business. We want to build a value system where people can scientifically validate their beliefs. So this is from
Thank you, Dr. Priti Singh. We have overshot the time. I'm sorry, there were a few comments that I would have loved to listen to from the experts, but I think we're going to be closing. Thank you all for your time. Ms. Dasu, Dr. Nandi, Dr. Rao, Dr. Singh, Dr. Sinha, Marani, and Thomas. And in case, we have just given the initiator, the idea, and if you have something that you can approach them, I believe they are available through media, through personal phone calls, or anything that you intend to do. Just keep in touch and approach them. They are going to be immensely helpful to any one of you. There are queries which can be very, very personal in terms of how you come out of this cell. We are in a particular silo where we don't want to leave that. So please interact with them. Thank you all so very much. And now, what is the next thing to be done before we come to the valedictory? Our volunteers to kindly take up the proceedings for the valedictory function here. Nimrata.
Former Vice Chancellor Nehru Shillong to kindly take the chair on the dais. Next, I would like to request Professor N. Saha, Dean, School of Life Sciences, Nehru Shillong, to kindly take the chair on the dais. I would like to request Professor S.R. Joshi, Convener and Head, Department of Biotechnology and Bioinformatics, Nehru Shillong, to take the chair on the dais. I would like to invite Professor K. Agwan, Faculty, Department of Biotechnology and Bioinformatics, Nehru Shillong, to take the chair on the dais. Now, I would like to invite Dr. S. Majal, Organizing Secretary and Faculty, Department of Biotechnology and Bioinformatics, Nehru Shillong, to kindly take the chair on the dais. With felicitation of the dignitaries on the dais, may I now request Professor S.R. Joshi to kindly felicitate our chief guest, Professor P. Tandon, with a bouquet and a hamper. I would request Professor K. Agwan to kindly felicitate Professor N. Saha with the bouquet and hamper. Thank you, sir. Now, I would like to invite Professor S.R. Joshi for the welcome address.
Very good afternoon to all of you. Honorable Professor P. Tandon, former Vice Chancellor, Nehru, and Chief Executive Officer, Biotech Park, Lucknow, a distinguished scientist, and a great teacher and mentor. I can tell you at the outset that what I'm here and standing here today, he definitely has a hand in making me to stand in front of him and welcome him. I welcome you from the bottom of my heart, wholeheartedly, sir. Thank you for accepting a person who has achieved so much in life can still remain so humble, and you are there as one example who stands out. Anything you tell him, he would never say no. And one such example is I have just made a request to be with us, and he drives here for this particular function. Thank you so much for your humility. And your humility stands definitely out. we can emulate in life to grow. Professor N. Saha, heartiest welcome to you, sir. He's our dean, a young dean, but anything that we approach to him, we'll just walk in and he'll make us to sit there and say, go ahead, you can give me the papers tomorrow. This is how we started our seminar. Thank you so much, sir, for all that encouragement that you have. Professor Akwan, my senior colleague in the department. The department is growing today. We have Professor Chatterjee. I would have loved him to be here because the formative years when the biotechnology department started, when the biotechnology department was inaugurated, it was Professor Tandon. As we mentioned, it was August 2007 when we saw the new building inaugurated when Professor Tandon was the Vice Chancellor. Then Professor Chatterjee was there as the formative head. He was the chartered head, I should call, of our department. Then it was handed over to Professor Rao, who is here, Professor S.R. Rao, who took a lot of pain in developing the department. Then came Professor Orwan, who took the mental in his head. And today it's on us, on me rather, I don't take it personally, it's all of us. And what you see today, it's all our effort the department has grown. I welcome you, sir. Dr. Sukti Mazao, my sincerest welcome to you, Ms. Mazao. And the greatest pleasure that I get is, when we talk here, I had a few students who have become successful entrepreneurs. Ms. Dasu is here, Amit was there, and Ms. Sukti was also my student. And what can be a pleasure that you can derive when getting somebody who is so young, energetic, enthusiastic, dancing with you, crying with you, walking with you, and sailing through together. Thank you, Mr. Sukti, for all that I welcome faculty from the sister department. We have head of the Department of Botany, my classmate, Professor Arun Kayan. Welcome, sir. We have Professor Vishnu Padaroy, my senior. I call him Vishnu Da. Welcome, sir, to this function. I have Arvind Bhaiya, Professor A.K. Singh. It's so nice to call you by the first name because you are very close, Professor A.K. Singh. Sumandidi, Professor S.K. Kumaria from Botany. Dr. Hiranjit Chaudhary, who is another student of mine, who is now with the Basic Sciences, Associate Professor there. Dr. Adana Bhattacharya, it's so nice. Again, he was once my student. We are colleagues now. Welcome, Dr. Bhattacharya, to this program. And my colleagues from the sister department, entrepreneurs who are here, the participants that we have are coming from far-flung areas, remote areas of Northeast as well as the mainland across the country. Faculty from different universities who are here, and our panelists who are here are chairpersons, co-chairpersons, media, and my dear scholars, the most important part of this function, I believe, and my MSc students. I welcome all of you to this particular function. We had wonderful 2 days of session. I know there are running around some last minute change, but I believe we enjoyed to our best. But before I finish my welcome speech, my heartiest welcome to all of you. I would like to put this on paper, my sincere apologies for any limitation and shortfalls that we might have had. And this was likely to be, you must have had certain difficulty where we are not able to be attending you or we are not available as scholars. Just take it that we are only 5 in number. And these students are hardly 30 to 35 in number with the scholars chipping in. And that was the human resource that we had. But we tried our best. But in case there were certain loopholes, limitations that we could not really come up to your expectation, you'll go back with, cherished memories that you live for long, forgetting the shortfalls. So just pardon us for all that may have happened. And I close by once again welcoming all of you to this very great function. Thank you very much.
Thank you, Professor Joshi. May I now call upon Dr. S. Majal to give this seminar summary.
Good afternoon, everyone. We have almost come to the final part of today's program, in fact, this 2 days national seminar program. and how the time really flied. I couldn't actually count on that. And we feel very fortunate that we have invited invitees and also invited speakers and participants coming all the way from different places. And we have actually tried, hopefully, we wanted to actually bring a platform where we could introduce to the participant a connection between academicians, a connection between those coming from industries, and also among the bio-entrepreneurs as well. So we started our national seminar on day 1, that is on 28th of February 2023, which happens to be also a National Science Day. And we had the inauguration program, which was inaugurated by our Honorable Vice Chancellor when it comes to the inauguration of the Bio-Entrepreneurs exhibition. And we had Mrs. Abhilasha Basayamot, who came here as our chief guest, and we have guest of honor, Professor Pramod Tandon, and also we had a guest of honor, Professor N. Saha, including the other dignitaries that were there, representing from different organizations of Meghalaya and the faculty from other departments of MEHU. So we started the first session by a talk by Professor P. Tandon, and he talked about, and his talk was focused on the rich biodiversity of Northeast region, the importance of bioeconomy and the era of bioeconomy in establishing raw materials on various sectors, floriculture as an enormous potential in terms of conservation and economic development of the region. Most importantly, he also cited contribution from his side in setting up the skill development and entrepreneur training in different regions of Northeast India that have greatly helped students in terms of knowledge, production, and expanding the scope, and not to forget his project in empowering self-help groups. Following his talk, we had a talk by Professor Dhruv Lal, and his talk focused on the importance of organisms and how important microorganisms are required in terms of startups, in terms of vaccine development, plant growth, and most importantly in maintaining the good personal health. He also discussed about the resistant bacteria coming from different environmental conditions, which are also an important area of research when it comes to microbiology. We also had a talk following him by Professor N.K. Dubey, and just having some reshuffling here, so sorry. So sorry. Yeah, we also had a talk by Professor N.K. Dubey, and he highlighted on the importance of underutilized plants, such as black rice, having a tremendous scope when it comes to exploring, and also the importance of ethnomedicines as well. After the talk, we actually divided into a different panel session, and one of the panel sessions that was running side by side was on bioiodine presentation by the participants who have submitted their ideas, and we actually had a committee or selection committee which were which actually came from we have Dr. S.K. Nandi, who actually came from Biomass Bioincubator Facility in Nevutura, and we had Sri W.F. Rani, who came from Prime Hub Startup Meghalaya, and then we have Dr. Malai Dey, who happens to be the consultant of State Council Science, Technology and Environment. So these three were the judges, or we can say the member of the selection committee who decided, and they have already given us their feedback and also announce actually the prizes. Although actually in the brochure initially we decided to give the cash prizes, but we also mentioned that ultimately it will depend on the suggestion and decision of the selection committee members, and it was decided that we are going to give actually the mementos along with the certificates with them. So in the following session, we had a presentation by Professor B.V. Mandal, and he discussed about silk as a substrate which can be used for tissue film formation, and he also discussed about the importance of 3D scaffold and bioprinting chip, which is a very good model preferred nowadays as an alternative to animal-based model, and he discussed about the importance of a bioartificial insulin as well. Following Professor B.V. Mandal talk, we had a talk by Dr. Chaitali Bhattacharjee. She discussed about the digitization importance, particularly during the COVID. There was increase in 40% of digitization, which has led to the accelerated development of vaccine, where she has given her insight on the innovation and the use of cloud computing, machine learning, and the quantum computing. Then we had a talk by Dr. M.K. Singhal, where he focused on the culture-dependent and culture-independent ways of analyzing foodborne bacteria. The process needed to be fast and specific, and they had used aptamers, oligonucleotide molecule for the quantification, and also he had cited the importance of graphene oxide nanoparticle, which could be an important approach as well. Following Professor Dr. M.K. Singhal, we had Professor S.R. Rao talk, where his talk focused on genetic diversity of different plant species, which include citrus, banana, and also cited that if plants, we know that plants have diversity, and it is understood genetically. If it is understood genetically, it will greatly help in formulating crop improvement and breeding program. So side by side, we had, as I discussed, we had panel session running in the other department as well, and where we had presentation by the participants, where we have discussed about their research findings in that. On the second day, that is 1st of March, we had a talk by Professor Chatterjee, A. Chatterjee, and just a minute again. So sorry. Yeah. On the Professor A. Chatterjee has focused. His talk was focused on the influence of heavy use of areca nut chewing and inducing cancer in the state of Meghalaya, and this was followed by a talk by Dr. R. Kuttu, who works on the application of machine learning and deep learning, and he efficiently showed how open source tools can be used to accelerate research. Then we had a talk by Dr. Preeti Singh. In her talk, she talked about immuno-oncology, which showed that the 3D clinical models are efficient in deciphering the complex tumor immune microenvironment. So we also had a parallel session, of oral presentation by the participant running in other department, where they elaborated on their research areas. We had a second session 5. This second session 5 was mainly meant for the entrepreneurs, and we have invited lectures from the bio-entrepreneurs. And the first speaker happened to be Dr. Swapnil Sinha. She elaborated how the pillars of entrepreneurial journey are important, and how creativity and leadership and passion is very, very important for an entrepreneurial journey. She showed us how one can convert the classroom competitiveness into a business zeal. Following her, we have Dr. Amrita K. Chaliha, where she took us on her success journey, which began with the correct identification of the market gap. Her all-female research workforce is a huge inspiration to all. Then we had a talk by Srimati Dasumarne Majha, Srimati Bajaj demonstrated her efforts to put Meghalaya on the map for all coffee lovers using locally grown coffee. She emphasized on the need of lab trials to achieve longer product shelf life and stability. Following her, we had a talk by Sri B. Marvin. Sri Marvin showed us how one can turn a crisis into a perfect opportunity, and he also showed how efficiently a barren land could be used, or a wasteland could be used to create an opportunity for people in rural area, especially for women. Following his talk, we had a talk by Amit Priyadarshan from Kaliche company, where he paved the roadmap from ideation to technology development in his talk entitled Northeast to Middle East, Our Journey of Entrepreneurship. He demonstrated how big risks are a part of a successful business opportunity. So following their talk, they were also having a side-by-side parallel session running on the other side of the building, where the participants were engaged in oral presentation discussing about their research findings. After this, we had panel discussion, and the panel discussion discussed about the problem and the issues that can actually generate in the process of when somebody wants to become an entrepreneur. And there were, we had a panel where we have representatives from the entrepreneur's point of view, we had representatives from the industry's point of view, we had representatives from the academic point of view. So we wanted to create a platform where there was a possibility of bridging the gap when somebody can come from academic line, wants to become an entrepreneur in future. So these are the overall summary that can I present to you now. Hopefully all the participants and particularly the invitees and the invited speakers have really gained knowledge and experience. And I would like to also mention special thanks to the bio-entrepreneurs. We had actually around 32 stalls set up by the bio-entrepreneurs, and we have bio-entrepreneurs coming from Assam, outside Assam, like we had from Mumbai and New Delhi. We also had many of them represented from Meghalaya as well. So out of these 32 stalls, they represented from a variety of background, and we are really happy that they were very supportive and they actually could come forward displaying the product, and I'm hoping that the students and the participants also, they could get benefit out of the interaction with these entrepreneurs. So with these few words, thank you.
Summary. May I now call upon Professor N. Saha, Dean, School of Life Sciences, for the presidential remarks.
Good afternoon to all those who are present in this hall. Professor Tandon is my teacher as well as former Vice Chancellor of this university. Professor Joshi, who is the convener of this seminar. Dr. Madhav, who is the organizing secretary. Professor Abu, so faculty of this Department of Biotechnology. Professor Rao, who is also a part of us, still I consider him still a part of us, although he is superannuated. Professor Kayan, Professor Singh, Professor Suman Kumaria, Chaudhary, Bhattacharya, Professor Roy, and other distinguished scientists who have come all the way from different parts of the country, and all researchers from NEHU as well as other parts of the country. Before I start, I would like to say something which I forgot to mention in the inaugural address. The hall which you are sitting here, I think this is the first time this type of seminar has been arranged. This is a community hall for the whole NEHU community, those who are residing in the campus. And this has been created or built under the leadership of our former Vice Chancellor, Professor Tandon. I hope We enjoyed very much having a seminar in this beautiful hall. I hope everyone will agree to it. It is due to his hard work, sincerity, we are using this such an excellent hall for organizing such a seminar. Even this exhibition which we are having here, behind that, the indoor stadium which you see, it is also the creation of a tandem under his leadership. And you see many of these, not only that, I cannot tell all, many of the constructions around in and around campus, it was done under his leadership. So we are very thankful to him to give so much of effort to have all these facilities which we are enjoying now. Okay. So now I come to this seminar. Although I am very sorry I could not attend these sessions because due to DGC visa, I had to attend various meetings as well as classes, so I could not attend it. But from the abstract I am seeing, there are about 14 invited lectures which are delivered by various distinguished scientists from all over the world, all over India. Then 35 oral presentations. And I can see 77 poster presentations and 21 BOP. What is that BOP? Oh, by presentations. So it is a really tough job to accommodate so many lectures, oral presentation within such a short period. And I was here till 7 o'clock in the department. Still, I came to know that these sessions are going on because one of the students who had to present, oral presentation, were there waiting. So it is really a great achievement. I would like to sincerely congratulate Department of Biotechnology in organizing such an excellent seminar. Especially the head of the department, under his dynamic leadership, along with all the faculties of the department, it has really added a new feather to the successful stories not only of the biotechnology department, also as for the School of the Life Sciences, because I take it with pride about, because I am a dean. So that's why I just added to it. It is an event which has been organized under the School of Life Sciences. Really, it is an excellent job. I could see the arrangement which has been made. I really like to sincerely thank the department, all the colleagues from the bottom of my heart. And then, you see, I'm sure, especially all your researchers, those who have come from NEHU as well as from other parts of India, must have been benefited from the all lectures in the field of modern biology given by various learned scientists or speakers or researchers. And then you must have also developed ideas how to do good research, how to do applied research, how to do research in a modern era. And then also you must have also acquainted how to use various tools and techniques. You see, tools and techniques is very important nowadays. You see, what type of technology you are using it. If you are really do not follow or proper technique you do not use it, your data will not be considered for publication. So that is why I am sure you also have got some idea about it. And then along with that, you see, this is I think, I would say for the first time which has been arranged. Along with that, the entrepreneur exhibitions was arranged along with this seminar. And it is such a nice display of all local products, okay, by the locals from Meghalaya, and how they really developed to utilize all these byproduct from this Meghalaya, some useful product for the human welfare. And I'm sure these researchers also must have got ideas how to do scientific innovation and ultimately become a good entrepreneur. And also, you can really start your own startup. You see, nowadays, it's a startup, era of startup. You see, Prime Minister of India is really encouraging to go for startup, you see. And then you can have lots of ideas about it. and you keep on thinking and all, and then lots of fundings are also being given in the name of startup nowadays. You see, I know there is one scheme called MSME. Okay, so this also you can just look at it. Government will give you plenty of money, and then not only that, you have to really, after finishing your study, you look for a job. You can also think to start something on your own. So these are things. I think it is all positive things. And lastly, I'm sure and I hope that those who came from outside Shillong enjoyed our new campus, the natural beauty of this campus, as also those who have got opportunity to go other parts of Shillong, you must have enjoyed it. This is all natural beauty, there's nothing artificial about it. Okay, so and then I wish all of you all the best and safe return to home. Thank you very much.
Thank you so much, sir, for your kind words. Now we'll be starting with the prize distribution for the various categories, starting with the biohydrogen category. As mentioned by Dr. S. Majal, the selection committee has decided that the ideas submitted by the participants are still in initial stage, which require more refinement. On the basis, the judges have decided to award the best 3 winning ideas with mementos and winning prize, winning certificates without the cash prize. I request our chief guest, Professor P. Tandon, to kindly give away the prizes, starting with the consolation prize.
Okay.
Mr. Arnab Chakraborty. Registration desk. Mr. Rakesh Ghosh. And that goes to Dr. Sanjayta Rajkumar. Which is a poster competition. Third prize goes to Ms. Lal Khansani from Mizoram University. Second prize goes to Mr. H.N. Maitai from Manipur University. Is Mr. H.N. Maitai present here? That will be received by Ms. Debarati Chakrabarti from Nehru. Our next category is Mr. E. Karmawalam from Nehu. Ms. Durba Nath from Assam University. Ms. Durba Nath from Assam University. Okay. And first prize goes to Ms. Joyati Das from NITGR. A big round of applause.
I consider myself to be privileged as I was present on the occasion of the inaugural ceremony and again at the time of valediction. I think this conference was with a difference. Normally one would go and listen to lectures, make oral presentations, posters. But here, the organizers tried to showcase what Northeast is all about. And I was particularly impressed with the stalls that were erected here by the entrepreneurs. The first stall, if I can recall, she has already given the description of presentations, but I think it's important to tell about the entrepreneurs also. The first stall, I think, Sally, you're a student who makes plastic ware out of waste. And I recall she was in St. Edmunds earlier when we had organized a training program. So she's continuing with it, which is a very positive development. Then house plants. Beautiful plants from here were displayed. Number of stalls showing the important spices, condiments, lakadong haldi, and so on. Bay leaf, cinnamon sticks, cinnamon powder. Not only this, you had the best of wines that people make here displayed. Then very sophisticated equipment to test the glaucoma in eye, which a gentleman, I forget his name, who did his master's PhD from Houston, and he has set up Malakman, Mr. Malakman. Then ladies from Institute of Advanced Studies in Guwahati, who specialize in softwares to detect cervical cancer in women using pap smears. Now, I may not have been able to provide you details of all the entrepreneurs there, but it gives you an idea as to what can be accomplished by people, and there is many more things that one can do. Then talking about the Bio Ideathon, where the participants presented their ideas, their business model, and so on. So, in totality, I think we have gained a lot at this conference, and it's a time to celebrate the success of this event, and I would expect all of you to give them a big hand. A big hand to Dr. Joshi, Dr. Majao, the entire Department of Biotechnology and Bioinformatics, and all those sister departments located here on campus: botany, zoology, biochemistry. As you can see that the teachers from all those departments are sitting here. I think such programs must be organized at least once by each science department. This is what I would suggest to our team, that kindly ensure that such programs are organized where people are able to exchange their research findings, people are promoted to enter entrepreneurship. Also, we need more startups, more incubators on campus, because whatever research we are doing must have some translational aspect to it, because when we do basic research, it's very important, but basic research eventually leads into commercial gains much later. But if you have a research which helps the society in promoting healthcare, in improving crop productivity, and so on, would have immediate direct influence on society. So I would wish to congratulate all the participants who have won awards, all those who participated at this conference, and let us say that we are going to have this time and again at this university and elsewhere. Thank you very much.
Now may I invite Professor K. Agwa, Faculty Department of Biotechnology and Bioinformatics, for a word of thanks.
Hello. Esteemed guests, participants, and organizers. It is my great pleasure to stand before you and express my sincere gratitude for your valuable participation in this incredible conference. Over the past 2 days, we have been privileged to engage in stimulating discussion, sharing cutting-edge research knowledge, and establishing new contacts with the colleagues from the different parts of India. As we prepare to close this conference, I am filled with a sense of pride and accomplishment for the outstanding work that has been presented here. The knowledge, expertise, and passion on display have truly been inspiring, and I am certain that the insights shared will drive new advancement, breakthrough in the field of biology, and ignite the young minds of new generation for bioentrepreneurship and benefit the humanity. The quality of research presented and the level of engagement demonstrated throughout the conference have set a high standard for future venues. On behalf of the organizers, I'd like to express my heartfelt thanks to the participants for their tireless efforts in putting together such a well-executed, informative conference. It is clear that their dedication and commitment in the advancement of biological knowledge have contributed immensely to the success of this event. I would like to extend my thanks to Honorable Vice Chancellor of NEHU for his kind consent to organize the conference, and the NEHU administration for providing the necessary facilities. I would like to thank Dean, School of Life Science NEHU for gracing the occasion. My deepest sincere thanks to all the resource persons, scientists, teachers who have spent their valuable time to be with us and sharing their knowledge. I'd like to thank all the bioengineers who have come from distant parts of country, particularly Meghalaya, to inspire the students for how to be a successful entrepreneur by picking up a societal need and meeting that need in a green way with their innovation. They deserve applause for their hard work. I'm thankful to all the sponsors of this seminar, starting from the Northeastern Council, Shillong, Science and Engineering Research Science, Board, Department of Science and Technology, Government of India, New Delhi, Northeastern Hill University, Bioresource Development Center, here in Shillong, Biolist Bioincubator Facility at NEHU, Tura, State Council of Science and Technology, Meghalaya, the National Academy of Science, NER Chapter, State Bank of India, NEHU Branch, Shillong, and FNDO. Without their generous financial support, it would not have been possible to organize this seminar. I would like to also thank Prime Hub Startup Meghalaya, which is our also sponsor. And I would like to thank the media houses, the T7 channels, the Meghalaya and All India Radio, and non-teaching staffs for their kind help and cooperation in making it successful. I would like to congratulate all the organizers, particularly the students of the Department of Biotechnology, for their tireless efforts in arranging each and every aspect of the conference and showcasing their hidden talent hitherto unknown by providing us with their memorable cultural nights. It was very sweet indeed. They deserve a round of applause. Finally, on behalf of the organizing committee, I would like to extend my heartiest and warm congratulations to Dr. S. Majab for our tireless efforts in organizing such a beautiful event. I would like to thank Professor Joshi, Head of the Department, Biology Department, for his support and all of his help in arranging the conference. Just when we did decide to hold the conference on 27th February and every arrangement had been done, suddenly election date was declared for the state of Meghalaya on the same day. It was a nightmare day for us. But thanks to Dr. S. Majab and Professor Joshi for their planning flexibility that led us to witness a successful conference. They deserve a good round of applause. Of course, I do understand that our invitees had faced some difficulties. We are once again thankful for your kind cooperation and sincerely regret any inconvenience caused to you. Once again, thank you all for making this conference an unforgettable experience, and I wish you all the best in your future endeavor. Thank you.
So we have, thank you Professor Aman. So we have come to the end of this 2-day national seminar. We thank the sponsors, bio-entrepreneurs, presenters and everyone else who has made this event a success. It was a great learning experience for all research scholars and MSc students. Thank you sir, thank you dignitaries who have been there throughout. We look forward to many such opportunities in the near future. May we now all stand up for national anthem and then proceed for tea.
For this Sufti Maza. Repeat. Hooray. Thank you so much everyone. Enjoy your refreshment and we are longing.
You take this one.
Bye for reading.
Why do you have to tell him thank you?